Ladies and gentlemen, please join me in welcoming Professor Molly Stevens from Imperial College, London. Professor Molly Stevens pursued her PhD in biophysical investigations of specific biomolecular interactions and single biomolecule mechanics from the Laboratory of Biophysics and Surface Analysis, University of Nottingham. Thereafter, Professor Stevens underwent postdoctoral training at MIT under the guidance of Professor Robert Langer. She joined Imperial College London in 2004 as a lecturer and was promoted to professor in 2008, making her one of the youngest professor ever in the institution's history. Professor Stevens is a professor of biomedical materials and regenerative medicine. Her research group uses innovative bioengineering approaches to pursue their vision of solving critical problems in regenerative medicine and biosensing. Their research spans drug delivery, bioactive materials, tissue engineering, biosensing, materials characterization, soft robotics, and the interface between living and non-living matter, and is underpinned by collaborations with data scientists and molecular dynamic experts. Professor Stevens is a fellow of 8 UK societies, including the Royal Society and the Royal Academy of Engineering, and the foreign member of the National Academy of Engineering. She holds numerous leadership positions, including the Director of UK Regenerative Medicine Platform, Smart Acellular Materials Hub. Please welcome Professor Molly Stevens.
Vesicles released from cells. People are also very interested in lipid nanoparticles, base nanoparticles, and also liposomes, as well as some other types of approaches. And on the right, I've just put a few papers that from our own group just in the last couple of years to show you that actually we work with quite a few of these different systems. And one of the things that was quite frustrating to us was the lack of techniques available to study these very tiny particles on a single particle basis to really understand their chemistry. And so we decided to actually invent a technology that would be the first one that would enable an automated measurement of chemistry of these particles at the single particle level. So this is a technique we've called Sparta, single particle automated Raman trapping analysis. This is now also being commercialized. And it's a technique where we can use an optical trap to trap an individual nanoparticle and then to measure its chemistry and to correlate this to its size and also to do some online dynamic reaction monitoring. And this is has all of our own software that powers this to allow automated trapping and release, and is now being made into an instrument that people will be able to acquire also in their own laboratories, so that you can really use this in a user-friendly way. And what we do is we trap individual particles, and then only if we have acquired a successful trap do we go on to do a high signal to noise acquisition. And if we don't, we would release the particle. And then we can go on and trap more and more particles and start to measure chemistry of these particles for hundreds, even thousands of particles at the single particle level. So you can pull out the kind of heterogeneity that you would never be able to see from conventional bulk-based measurements. So I'll show you some of the things that we can do. We can measure, for example, different types of liposomes. At the top, we have some deuterated and some non-deuterated liposomes in the green. And without having to label these with any labels or anything, you can distinguish between those two populations if you look at a mixed population with the Sparta machine. Similarly, you can do this sort of work for polymer-based particles as well. So at the bottom, we have some polymersomes. They're the same chemistry in the blue and the red there in the bottom left, except the blue also has an additional heparin group along the coupled to the polymer, and you can see that appearing in the Raman spectra. And again, this enables us to distinguish between these two different chemistries without having to label the particles. And you can also correlate that to the size of the particle. We do that by allowing the particle to displace a Raman active solution marker when it moves into the optical trap. And so we can, for any given composition of particle, we can work out also its volume. And that's really interesting in terms of thinking about mixed populations where you might have different size particles, and you would be interested in how that different chemistry, for example, might occur with different sizes, but also different types of drug loading and different interaction with things like the protein corona. And you can also measure reactions in real time. So what we're looking at here is an experiment where we have a polystyrene particle in the top left there, and we can then undergo a chemical reaction to introduce an alkyne group, and this shows up in a very characteristic peak in the Raman spectra. And then you can go through a subsequent reaction to convert this to a triazole, and you then will see the disappearance of that alkyne peak, but the appearance of a triazole-based signature instead. And at the bottom there, you can see how we've tracked this on single particle over time. So you can actually monitor chemical reactions on single particles over time, which is something that's normally very, very difficult to do. We also are really interested in how we might apply this to look at exosomes. And for example, we know that exosomes are released from cancer cells, and they have a very different signature when they're released from a cancer cell compared to a healthy cell. And so by being able to trap these exosomes and monitor their chemistry, we've been able to distinguish between exosomes that are originating from cancer-based cells versus non-cancer-based cells. And this gives us a really high sensitivity and specificity with this approach. And if you're interested in this, we've looked at now many different types of exosomes, and these are published in this recent paper in ACS Nano. Now I know that Professor Banerjee was very interested in lipid-based particles as well, and so I wanted to show this very recent example, which is from an Angevante Chemie paper that we published, was accepted actually just a couple of weeks ago. And what we're doing here is using Sparta to look at the particle composition analysis for single lipid nanoparticles with different chemistries, and also very interested in how these can be essentially partly digested by phospholipase enzymes. And these kind of enzymes, in particular here phospholipase D, occur naturally within the body, and they're associated with some particular diseases. And what we're going to be looking at is the conversion from DOPC to a removal of the head group. So we can look at the composition of different types of lipid nanoparticles to start with, and we can then measure the dynamic reactions as these enzymes interact with them. And this is the first time this has ever been done for single nanoparticles. And it's really important, actually, if we think about understanding these systems so that we can make the most use about designing better lipid nanoparticle-based systems. A number of these, actually still very few in number, but some of these have now reached clinical use, including in some of the Moderna vaccines. And there's a huge, bright future for these. And I think for different disease types, we want to be able to essentially design these systems as well as possible. So what I'm showing in this graph here is just how we can use this Sparta Raman spectroscopy to measure differences between these different lipid compositions in the particles. In particular, you can see quite dramatic changes in a peak that's related to choline. And then as you change the different composition, As shown on the right there, you will see these different ratios between the choline to cholesterol Raman peaks, and this enables you to pull out the signature for particles that have a different kind of composition. And you can then also monitor how these types of particles are affected by the presence of this enzyme, phospholipase D, which will cleave off part of the head group. And so in the left here, we're taking single particle measurements for different concentrations of phospholipase D when we incubate particles with this enzyme. And what you can see on the right is that the time for the enzyme essentially to cleave off about 50% of the groups changes quite a bit depending on the enzyme concentration. There's also a lot of very interesting mechanistic things that I won't cover today, but that are reported within this Angivante paper. It's very recently accepted, just it's still in uncorrected proof form, but you can find it on the website if you're interested in that. Sorry, can everyone mute their microphone, please? Thank you. Okay, so we believe that this Sparta technology can be applied to help in formulation development and manufacturing process development, as well as in final QC applications as well. So looking at the quality control of the sort of nanomedicines that you would make.
Everyone is requested to please mute their mics. There's some disturbance.
Thank you very much. So we can look at a number of different particles. I've shown you some examples here for liposomes, for lipid-based nanoparticles, for polymerasomes, but we've also looked at other particles such as viral vectors. I've also shown you a little bit of our application in exosomes. And we can look at composition. What I haven't shown you today, but we've done quite a bit of work of, is looking at things like drug loading, and we can look at these in a number of different formulation buffers. So I hope this kind of technology would be useful to a lot of you, and please do get in touch if you're interested in doing some measurements with this, either in collaboration with us or through also perhaps acquiring some of these machines in your own laboratories. I want to move on to show you a little bit now about how we can use very small needles to help us with drug delivery. There's quite a number of different physical triggers that we use in our group for drug delivery. We've used ultrasound a little bit. We are also working quite a bit on using light, particularly with activating CRISPR-based systems for gene reprogramming. But today I'm just going to show you about some mechanical interfacing that we've done that achieves very good drug delivery. And there are a number of technologies, nanotechnologies out there that can be used to interface with cells. You can see some of these in this particular schematic from our recent paper. And this is really nice technology, but it's a little bit difficult to apply in vivo. What we've been working on are these beds of very tiny needles that we can use to deliver molecules into cells and also to measure what's going on within cells. And I won't have time to cover too much about the chemistry of these today, but we have published quite extensively on these, including this recent review paper here in Advanced Materials that covers work from our group and also from other excellent groups in the field, looking at the different kind of applications you can use these for. So delivering molecules for therapy, using them to stimulate cells for mechanotransduction, also using them for biosensing and also bioelectronic applications. This shows you a cross-section of a cell on the needles. And what we see is that the needles go very closely inside the cell, but they never penetrate the nucleus directly. They're just getting close contact, as you can see here. And we can use them to deliver lots of different cargo. So this was from one of the first papers we published, where we used them to deliver molecules that could, molecules called VEGF plasmid DNA, so for VEGF-165, which is plasmid DNA that can encode for vascular endothelial growth factor. So it basically makes the cells produce this growth factor that then means the cells produce new blood vessels, which is what you can see in the image on the right. So we put these needles onto muscle, living muscle in the body, remove the needles, and then the delivery of the molecules means the cells are then able to make new blood vessels. But you can use them to deliver many other things. And one of the things we've been using them to deliver are very small nanoparticles, as you can see here. So in this particular example, we've used them to deliver quantum dots into cells. You can see in the top left, the quantum dots show up white in the electron microscopy. They're inside the needle, but they also get into the cell. And we were excited by this because our group was the first group to develop quantum dot-based nanoparticles for kinase and acetyltransferase activity biosensing. So we've always been interested in how can we get nanoparticles in a better way into cells, and this needle approach enables us to do that. And that really takes me to the second thing that I want to talk about is how we can use nanoparticles for biosensing. And what you can see in this schematic here is different ways that you can think about functionalizing the surface of nanoparticles. We've worked a lot with looking at how we can make nanoparticles responsive to enzyme activity and also to detect different types of nucleic acid structures, also to detect different types of proteins. And so there's a lot of work going on around the world to enable people to use nanoparticles in interesting ways. And this is relevant really for pretty much every disease you can think of. So detecting early heart failure, for example, early cancers, antimicrobial resistance, which is something Dr. Banerjee was very interested in. Also, of course, early infectious disease detections. And nanoparticles have been around a long time. You can see some of them here. These are gold nanoparticles within the walls of the Lycurgus cup, which is dating back from the 4th century AD. But it's much more recent that we've been able to really control them. And so what I'm going to be telling you about today is a couple of examples where we've used nanoparticles and we've thought about their surface and their biosensing capability to use them as potentially for drug screening, but I won't talk about that today, but also for early disease detection. And we also collaborate a lot with Professor Jarowski for all of the molecular dynamics that you can see on this slide. So first, if you wouldn't mind muting your microphone, please. First example that I can see tell you about here, perhaps the chair can also mute the people that are unmuted so that, yeah, thank you. Yeah. I'm going to show you this example here about using nanoparticles to detect cancer early in the body. And we are using nanoparticles that can detect these enzymes called proteases. They can cleave through peptide chains. And the idea is that we will get our nanosensor used inside the body so that our nanoparticles will end up in the urine and will then turn the urine blue if the patient has a particular cancer there. And the biosensor you can see is in the top left here. We have a protein core, and then we have some very short peptides that are attached to this through a biotin neutravadin linkage. And then the short peptides have a very tiny gold cluster on the end of them, which is about 2 nanometers in diameter. But that whole complex is 11 nanometers, and that's too big to go out through kidney filtration in the mouse. And so what happens is when you inject this complex into the mouse, it will not come out through the kidney at early time points unless you have an upregulation of the enzyme related to the cancer. And that enzyme can cleave through those short peptide chains, release the gold nanoparticles. And because these are now only 2 nanometers in diameter, they can come out into the urine, and you can add some chemicals to that, and they will turn the urine blue. You can see here the particles and here the actual data. This is from a mouse model of colorectal cancer. The in vivo work is done in Professor Batia's group at MIT. And you can see that with the healthy mice that don't have an upregulation of this enzyme related to the cancer, which is called matrix metalloprotease 9, the urine stays yellow, but in the cancerous mice, the urine turns this sort of very bright greeny blue color if you collect the urine after 1 hour after delivery of these particles to the mouse. So this is just a proof of concept, very early preclinical data, but we're interested in taking this forward for a number of different applications. The last thing I want to tell you about is some of the work we're doing in trying to help with democratization of access to healthcare. In particular, we all know that infectious disease really disproportionately affects lower income countries. And so there's a lot, I think, that can be done to help with getting technologies that are more accessible around the globe. And in particular, we're interested in how we can use technologies that might be able to be read by mobile phones. Now, the reason that's important is that there are so many global mobile phone subscriptions now, in fact, 7 billion across the world. And if you look at even smartphones, and this is from a paper a couple of years ago we published in Nature, if you look even at smartphones, even in sub-Saharan Africa, the use of these is increasing quite a lot, as it is all around the world. And the capability of those phones is also increasing a lot. And so the phones themselves can take better and better images, for example. What we looked at here was a mapping in Uganda of patients that would be within 5 kilometers of a healthcare facility versus 5 kilometers of access to network coverage to use diagnostics that could be read by smartphones. And what was very striking is that there are really quite a lot of patients that might find it quite difficult to get to a healthcare facility, but would be much better served by having access to a diagnostic that could be read locally by a smartphone. So we set up this center a few years ago now, actually before the pandemic. It's called iSense. I'm the deputy director. We look at infectious diseases. We track symptoms within populations. This is done by Professor Ingemar Cox at UCL. He works a lot with Google and Twitter as well. I work on the ultra-sensitive biosensing test that can be read by mobile phones, and we then look at integration into online care pathways. And we're working on many, many different systems within iSense, but I'm going to show you just a couple of them, which are really based on ultra-sensitive biosensing. And one of the things we wanted to do was to detect HIV much earlier at the point of care, and to do that by detecting the virus itself. And by detecting the virus itself, we're looking at a protein on the surface of the viral capsid called P24 that you can see in the image here. And the reason we wanted to do that is that normally at the time we did this work, which is about 3 or so years ago, people were only able to detect at the point of care by actually measuring antibody response to the virus, which would take several weeks. So you were missing out on early detection. And we've developed a number of different types of chemistries that can be really useful in enhancing the ability to do very ultra-sensitive biosensing. And what you can see here is just one of these technologies where we have a gold core with a porous platinum shell. And these are particles that can essentially act as artificial enzymes. And so if they are present, they will turn, if you add some substrates to them, they will turn this very kind of dark bluey black color. And you can use these in a lateral flow test, similar to how you would use a gold particle, for example, in the lateral flow COVID test that you will have all seen, but they can give a much stronger signal, so you can get a much enhanced amplification of the sensitivity. You can functionalize them with antibodies that can recognize the P24 protein from the HIV. And then we can use the patient sample, and into that we also add a very tiny nanobody, which can also recognize the P24, and this has a biotin on the end of it. And this can mix with the P24 from the patient, and the P24 will sandwich between the particles and the nanobody, and the mix can then be run up the test strip, and it will bind to streptavidin at the test line. And because biotin and streptavidin can bind very strongly and very quickly, this gives us a very efficient capturing of particles at the test line. And then we can add some chemicals so that the nanoparticles can oxidize this peroxidase substrate and generate this colored product. So the test line changes color. And this really gives us 100-fold amplification. So it's a huge enhancement of signal, much more sensitive than the traditional gold particle-based lateral flow assays. And you can see, this is back from our paper in 2018, the dashed line there is where the commercial gold standard kit from Alere was performing, and we are in the red, the data points there after amplification to get us right into the acute stage of HIV detection. In fact, sub 1 in terms of concentration. So really, really sensitive early detection in a point of care format. We're working very closely with the partners in many different countries around the world, but including in, this is from a recent trip we took to KwaZulu-Natal, where our partners at the Africa Health Research Institute also work a lot with local communities. And I'll just show you this video here, which is from iSense, which shows you some of the work that's been done within iSense around making also these readers that can capture tests and have facial markers in there, so that when you take photographs of these, you get very robust results, and you can then map these in real time onto data dashboards. And a lot of this work is in collaboration also with other professors within the iSense consortium. It's a really fantastic team that we have working together there. And then very finally, what I want to do is to show you this application here, which is one we took to Uganda for measuring Ebola virus antibodies in survivors of Ebola. So to distinguish between which of 3 different strains these survivors had in the Gulu outbreak. And this was tested on close to 100 survivors, and the test that we developed could distinguish really well between those different strains of virus based on the serological testing of the patients, and you can also geotag these. Now you'll see this is back from a few years ago now, and this kind of approach really, I think, is becoming more and more widespread now, thanks to the pandemic, which is great to see, and you know, is almost now becoming well, hopefully will become really routine, because I think it's really important in terms of really understanding spread. And it's great to see that, because when we published this back a few years ago, you know, this is certainly very far from routine. So I'm just going to finish off just reminding you again the kind of different strategies that we use in our group around the biomaterial interface for a number of different therapeutic areas. I've very much today focused just on topics that I felt were really pertinent to the commemoration of Professor Banerjee. But I do hope that you know, this is of interest to people in the audience, and again, really join you in the commemoration. And thank you so much for including me in this.
Thank you, Professor Molly, for your wonderful talk. I will be the moderator with the Rajkumar for this session. And I request and welcome the participants to ask any question if they have to the Professor Molly. We can take few questions for sure. Ankur, Professor Ankur, you can ask questions.
Yeah. Molly, fantastic talk. Really enjoyed it. I have a question related to your microneedle delivery system. Have you thought about using the system to perturb your nuclear component? So it penetrates through your membrane and goes into the cytoplasm, right? But you can probably fine-tune the length of those needles to go and poke into the nucleus side and perhaps play around with the chromatin structures and all. Have you ever thought about or done anything along that line?
Yeah, it's a fantastic question. So actually, even in terms of the cytoplasm, there is still an outer cell membrane on the needles. It just becomes very permeable, but it is still there. We see very clearly the nucleus deformation, and we've published a couple of papers on that. One by Katie Hansel in my group looks at changing lamin ratios within the nuclear membrane. We also know that we see some interesting, mild but interesting epigenetic changes in the cells that relate to a better ease of reprogramming as well with that kind of perturbation. So I'm really interested as well in thinking about how we can do even more imaging with fish and techniques like that. We haven't done that yet, but I think also understanding much more about chromatin structure, as you suggest, is something we've definitely been discussing and thinking about.
Thank you.
Rajkumar, would you like to ask a question?
Hello, ma'am.
Hi.
So I have heard your exosomal research. So recently we also started actually exploring exosome research. So you have told that exosomes can be extracted from cancer tissues and everything, but also at the same time we have studied that exosomes are can be used as a therapeutic vehicles for drug delivery. So at the same time, there is also controversial reports that exosomes promotes pro-inflammatory secretions in terms of like promoting inflammatory things. So in the future, where do you think that how we can explore what kind of techniques we can use to explore the whether it's pro-inflammatory or else anti-inflammatory, because many different diseases also can be used, like fibrosis and also other inflammatory diseases.
Yeah. So our technique that we showed today, the Sparta, really is about profiling differences in the chemistry. So you would always want to couple that, to some in vivo studies, for example, to look at structure function outcomes. But if you're interested in general, we have written a few perspectives around exosomes and their use in therapeutics as well, including actually publishing a little bit of work highlighting some of the things you need to be careful about around some of the artifacts of when you use these systems. And I do think that they have a lot of really interesting therapeutic applications, but there is quite a crowded literature at the moment, as you've probably seen, and there's also a number of artifacts within the field. So I think it's really important that we kind of standardize how we look at these particles so that we have better characterization, so that when different labs and then studying them in different assays, we can, cross-compare in the most robust way.
Thank you, ma'am. Thank you very much.
Thank you. Actually, the nanoparticles and nanoparticle combination interests me a lot. So I wanted to ask the question that what kind of application, multi-application delivery can we use? Like, could you give us more insight about the application that it can be?
Yeah, so you can use for a number of different applications because they're very short. Really, it's most applicable for applications where you can access the surface that you want to treat, for example. So some of the previous work we've done, as I mentioned, is on accessing muscle, but we've also looked at a number of applications for these where we use these in the GI tract, for example, and also we have a lot of work going on where we're using them for cardiac applications. So, You can think about using them for delivering therapies or nanoparticles, as I mentioned, or also elements that could help with gene reprogramming. And the other thing we can do, I think that's pretty interesting, is to use them for biosensing. So we've used them quite a bit to look, for example, at distinguishing the chemistry of cells within tumor margins, so that you could apply these. The idea is you could apply these during an operation and use them to help label your cancer versus your non-cancer cells. But really, it's for applications where spatial control of delivery is something that you're going to be interested in.
Thank you, ma'am. Thank you, sir.
Thanks, Lisha. Does anyone have any question? Or okay, Dr. Molly, I have a small question regarding the Raman-related technology. Have you tried to characterize the gas-filled vesicles using the technique which you have commercialized recently?
We haven't. Do you think that would be interesting?
I think because there is a gas escape happens from the liposomes or even polymeric mesh, so it would be. I think it would be interesting to learn how it is operating by using the technique which you have developed.
Yeah. Okay. Maybe you can email me afterwards.
Sure. I will do it.
But that's not something we've looked at or thought about looking at.
Sure. I will email you. Thanks. So anyone has any questions, or shall we go to the next speaker? I guess no one has any questions. Thank you, Dr. Molly, for your wonderful talk today.
Thank you so much.
Thank you, Doctor.
Thank you.
Thank you.
Ladies and gentlemen, please welcome Dr. Ankur Singh from Georgia Institute of Technology. Dr. Ankur Singh is the Woodruff Faculty Fellow and an Associate Professor of George W. Woodruff School of Mechanical Engineering at Georgia Tech and Wallace H. Coulter Department of Biomedical Engineering at Georgia Tech and Emory University. Dr. Singh pursued his PhD in biomedical engineering from the University of Texas at Austin. He continued his postdoctoral fellowship in the Department of Mechanical Engineering, Georgia Tech. Dr. Singh is an M.Tech alumnus from late Professor Renti Banerjee's lab. He has published over 60 articles in peer-reviewed journals of repute, including Nature Methods, Nature Materials, Nature Nanotechnology, Nature Immunology, Nature Communications, PNS, Blood, and Advanced Material. He has written multiple editorials for Science Translational Medicine. His work on immune organoids were identified among the top 100 discoveries of 2015 by Discover magazine. He is the founder and past chair of the Immune Engineering Society for Biomaterials and Controlled Release Society. With these words, let us welcome Dr. Ankur Singh from Georgia Institute of Technology.
Dr. Ankur, can you share your presentation, please?
Yeah, hold on a second.
Thank you.
Can you see my screen?
Yes, I can see your screen.
All right.
Thank you.
Well, thank you for the kind introduction and the invitation to speak at this important memorial meeting for Professor Banerjee. For those who don't know, Rinti had a had a major impact on my career. She introduced me to the field of biomedical engineering and was my MTech mentor, advisor at IIT Bombay from 2004 to 2006, and really shaped my career because before that I was thinking of much more aligned with different tracks for my career trajectory, and working with her really inspired me to get into academia, pursue. Uh, but it's very difficult to even accept that we don't have Rinty anymore, and um, she was a wonderful mentor person, scientist, and she gave me an opportunity when I came to Bombay. It was she gave me an opportunity to work with her. We published together, a whole patent together, but in many aspects, the relationship that I developed with people there in the lab as friends, but also I found my future wife over there, Dr. Shalu Suri, who was also Rinty's master student, and we continue to talk about the impact that she has left on our lives. So today I want to share some of the work that my lab has been doing, which has inspiration from what I learned with her during my master's, which was in developing materials-based technologies for cancer research at that time, and how it has impacted the work that I do now. So my lab has several key areas. We work in the area of infectious diseases, from generating antibodies in a dish and organoid development to vaccine developments. We work in areas of cancer research, primarily lymphomas and prostate cancer, inflammation of arthritis and skin, and more recently, metabolic syndrome, immunomodulation in the context of gut microbiome. My lab goes from engineering and technology buildup to understanding fundamental mechanism related to the immunology, and eventually leading to therapeutic development. So some of the aspects that I'll talk today are related to development of this technology called immune organoids or lymphoid organoids. And then the later part of the talk will transition into an upcoming work, which is about creating nanoneedle platforms to manipulate the fate of T cells. To introduce you to organoids, these are in vitro miniaturized culture platforms that have been developed for a large number of various organ system at this point, primarily from the stem cell side or cancer sides, and they work through various self-organization of the cells, where cells come together and form a particular structure that are representative of selective aspects of how your tissues are organized, or your organ systems are organized. You start with various progenitors, undifferentiated cells or inactivated cells, and give them the right cues. Those cues can come from a variety of signals that you can provide, from materials to cytokines to growth factors, and a lot of other various factors that are there. My lab's focus has been on a particular organ system, which is the lymphoid organs. So in that, we are focused on secondary lymphoid organs. So you have a primary lymphoid organs as bone marrow, thymus. The secondary lymphoid organs are where the immune cells undergo activations. For example, your lymph nodes, spleen, tonsils, which has been some of our more recent progress in that area. And we're interested in building these lymphoid organoids in dish or on chip using microfluidics, so that we can understand the decision making of these cells, what makes them start the immune reaction, what makes them decide it's time to stop, move forward. To develop immune therapeutics in a dish, for example, generating antigen-specific antibodies in a dish against previously unknown infections. And this also converges into a big part in my lab, which is lymphomas, which are the tumors of the same immune cells, such as B cells, which undergo wrong mutations, in simple words, and get locked in a state where they cannot be eliminated from the body, and they turn cancerous, as shown over here. So we're interested in developing these organoid technologies so that we can understand the oncogenesis of these tumors, the drug resistance, what are the drivers of this, both at the epigenetic and the signaling level, and develop new therapies. To introduce you to more about what we do in the organoid space for the lymphoid system, we're heavily focused on the B cell immunity aspect, which is the antibody making system in our lymph nodes. So here's a section of the lymph node where you have a subcapsular sinus through which the antigen is transported, and next to that are these B cell follicles surrounded by T cells in the outer zones. And the antigen can be transported into these B cell follicles through a variety of mechanisms, as soluble antigens or nanoscale vaccines. And there are some very interesting work out in the field that you can read a little bit more about this in this review article that I had published. What happens at the B cell level is that in response to the antigen that's presented to the B cell, you have an activated T cell, which is also an antigen-specific cell. And when this interaction takes place, one of the key molecules that are involved are CD40 ligand. And with that interaction, the B cell has a couple of options to do. One is to undergo a short-lived plasma cell formation, very rapid response, but leads to often low affinity antibodies. The second is to undergo this classic reaction called germinal center reaction, which is a stepwise process, very controlled process. In this, in response to the antigen and the T cell interaction, the B cells undergo massive proliferation, followed by diversification of its repertoire, then class switching recombination. These things happen in a zone called dark zone because it is optically dense. From there, it migrates adjacently to a light zone where it undergoes a selection process. Here, you have a couple of immune cells, such as a specific type of T cell called follicular T helper cell and follicular dendritic cells that give the right kind of signals and interact with these prime B cells to tell them whether they're good to go forward by recycling the process and improving, or should be eliminated. The end product of all of this through repeated recycling is generation of memory B cells and the long-lived plasma cells, which tend to protect us over the long term. So fairly complex process, but it remains quite a bit of a black box in the field of immunity. And is of interest to us. To understand this better, when you have B cells that are exposed to antigens, a lot of these clones that are there for B cells, they undergo competition. So imagine this as a Darwinian approach where there are a lot of competitors over there, but the winners are going to be fairly rare population. So using this beautiful model that Gabriel Victora's lab has published in Science, a lot of these clones compete with each other, and then they segregate into these individual colors. This was done using a confetti mouse model, and the winners tend to be the ones of a specific clonal type, and the rest of the cells should be eliminated. But whenever we take these pictures, we're looking at a very static image, but this is not how it happens in a lymph node. So for example, this is a video we published on 3-photon imaging recently on germinal centers and T cell responses. And you'll see how these cells, these are the B cells undergoing decision making, and they are really migrating inside the lymph node and the B cell follicle to make these decisions. Eventually, one of these colors will become the winning population in this that protect us from infection. generates immunity against vaccination. So how do we do this outside of the body? There's an elegant work done using mouse models and non-human primate models, and also sampling human population with those. But how do you do this outside? So we went back into a classical approach where a stromal cell has been developed by a group in Japan from Daisuke Kitamura's group. And this stromal cell is a fibroblast, basically, that presents the CD40 ligand, which is a T cell signal. And when you take a naive B cell and incubate with this in a two-dimensional culture, you expect the reaction to take place. Unfortunately, you have very limited response in terms of the proliferation of B cells, which is a classic hallmark. They should be undergoing a lot of proliferation, but you don't see that much. And very few cells actually undergo classic changes that are associated with the gene expression level of the germinal center. So for example, if you have a naive B cell over here, in vivo, you have a lot of AGDA, which is one of the important drivers or indicators of germinal center B cells, highly expressed, but expressed at low levels in two-dimensional system. And likewise, many others are either up or down, but they're not really close to what might be happening in vivo. So we took a step back to understand what happens really inside the lymph node, and we collaborated with Dr. Jason Sister at UCSF to look into some of the single cell sequencing analysis and some of our own analysis of lymph node proteins, such as the extracellular matrix proteins and genes involved in binding to those extracellular matrix. And we found that various stromal populations, such as follicular dendritic cells, which is very much present in the B cell follicle, expresses a lot of these genes that are cell binding proteins that encode cell binding proteins, such as VCAM1, which is vascular cell adhesion molecule 1, collagen 1, vitronectin, fibronectin, but not hyaluronic acid or laminin. When we look into the integrins that bind, we see specific integrins that bind to some of these collagen and vitronectin moieties. So we confirm that what might be present in the two-dimensional microenvironment. This also has some inspiration from Dr. Darrell Irvine's work from 2000s, where they had developed some of these technologies to evaluate immune cell migration outside of the body. So how do we do that? So one of our first system was using a nanoparticle-based system in which we took gelatin as a starting material, and we used silicate nanoparticles as just taught and as ionically charged particles to cross-link these gelatin-based structures into a hydrogel platform. And within this hydrogel platform, we had the stromal cells that were presenting the T cell signal and secreting key factors that would drive this reaction. The output of this is that you have a formation of these hydrogel-like structures, which you can give any shape you want to that. We mostly made disc-like structures and also matched the mechanical properties of these lymph nodes that we had determined using some micropipette aspiration assays. As an outcome, when you take these B cells that are encapsulated as single cells along with the stromal cells on day 0, by day 3, you have these self-assembled clusters that are formed within these hydrogels. Therefore, we call them immune organoids. And these cells that are undergoing differentiation, they start expressing this protein called GL7. These are mouse cells. Mouse B cells freshly isolated from the spleen and the lymph nodes, and they start undergoing differentiation. By day 4, we see about a 100-fold increase in the expansion of these cells compared to a very limited expansion of the phenotype in a two-dimensional culture. The important part is that not only you have a massive increase, but you can actually fine-tune this or control this by changing the ligand density or the presentation of number of these supportive cells or the biomaterial properties in this. But this is a phenotype. This doesn't really tell you that you have anything close to a true germinal center reaction other than a protein expression. So we did extensive analysis on the whole transcriptome and somatic hypermutation, which are the key hallmarks of germinal center reaction. And a very surprising and exciting moment for us was when we found that the organoids were remarkably close to the transcriptome of the in vivo germinal center B cells. So here we immunized a mouse, isolated the cells from the immunized mouse from the lymph nodes, and compared that to those that were derived from the organoid in a dish. By day 6, we actually are moving into a plasma cell state, which is a more advanced stage of the differentiation. So this data is further shown over here through, and you can see how remarkable the day 4 gene signature is compared to the in vivo. The second is the somatic hypermutation that allows you these cells to have the VDJ recombination on them so that they can be specific to a particular antigen type at some point. And this was something that was also quite close in the organoid platform compared to the in vivo. So likewise, we validated many such hallmarks, and we found that the three-dimensional system was quite close to the in vivo system and quite separated from a two-dimensional presentation of that. And there can be many reasons for that. We're still exploring that. Some of those could be the better performance of these B cells because of the matrix that we had provided, or the secretome of the cells that we're using as support cells. So while we're interested in on the vaccine side, we were also interested in solving a standing question in immunology, which is when you get infected or vaccinated, your germinal center B cells massively proliferate. So what really controls this massive pace? And if we can do that, then we can understand how to design better antigen vaccines or adjuvants so that we can expedite the vaccine response process, of which we have seen in the past 2 years how the dire need to develop vaccine in a rapid fashion. So our focus in this study was on the epigenetics, which is at the chromatin level. There are several on and off switches found in the B cells that have been reported in the literature, such as EZH2, which is a histone methyltransferase, KMT2D, CREB-BP, and EP300. Some of these switches are on, some of these are off. What means is that they are important for the progression or termination of B cell maturation process. Or using some human tonsil system B cells that were isolated from them and performing some chip sequencing, we came up with a hypothesis, which is that EZH2 is a master regulator of the striking germinal center proliferative phenotype. And this process is mediated through a cell cycle checkpoint gene called CDKN1A. EZH2, what it does is it trimethylates histone 3 and allows the germinal center reaction to progress. So to test that, we collaborated with Dr. Ari Melnick at Cornell Medicine, which was my previous institution. And there we developed first the mouse models, transgenic mouse models. So first, this is a wild type. If you just focus on this particular panel, this is germinal center in the spleen of immunized mice. So you get a nice germinal center formation in a wild type. You get nice germinal center formation in a CDKN1A knockout mice, but you do not get any germinal center formation in an EZH2 knockout mice, which tells that EZH2 is very critical for the formation of germinal centers. But when you cross these two mice to create a double knockout mice, then you rescue the response of germinal centers, which is quite surprising, but was supporting our hypothesis. Quantified over here, you can see that you barely get any germinal center formation in the EZH2 knockout, but the double knockout of EZH2 and CDKN1A rescues this process. So how do we study the cell cycle in this? The problem is that the cells undergo migration within this structure. This is a transient structure. They undergo migration. All of these cells can have different cell cycle states. So how do you control that in vivo? And the second is you don't have any cells over here. So what do you have to? You don't get anything to study further, such as cell cycle states and all. So to do that, overcome that, we developed the organoid platform. We modified it to fit the need of the question. And we first validated the epigenetic signatures in these. So for example, EZH2, which I've emphasized so far, is in vivo highly expressed, and in ex vivo in the organoid platform is also highly expressed, but not in the two-dimensional culture. So again, proving that this is the right system or closer system, I would say, to do that. So likewise, various other cell cycle molecules are better expressed in the three-dimensional system that they are expressed in the 2D system. So what we did over here was we now took the organoid platform and took the freshly isolated naive B cells from all our mouse model, the 4 system that we were using, and we wanted to see, can we even reproduce this in these transgenic mouse behavior in an organoid platform. This was a wild experiment, and we were super excited when we saw that the pattern that you see over here in vivo with the wild type CDKN1A knockout, EZH2 knockout, and the double knockout was remarkably reproduced ex vivo in this organoid platform that you see. This is the germinal center phenotype. So long story short, what we did in this paper was published in Nature Communications a couple of years back. The cartoon summarizes the detailed finding of this paper that when B and T cells interact with each other, they undergo uphill, where several transcription factors are involved, which we validated using bone marrow transplantation studies as well. And at the top of this reaction of germinal centers, you have EZH2 interacting with CDKN1A. When you take the bricks off by manipulating these, then you get rapid proliferation that allows the B cells to proliferate, diversify, and the process continues to the recycling until you have a winner cell population that comes out. The highlight is that this research was not possible without making those organoids, because years of our effort into the mouse model alone had failed. And when we did that, that's how we were able to prove this point, which emphasizes the need for a biomaterial-based technology that can drive this process. Now, as I showed earlier, through the sequencing, RNA sequencing approach, that there are various adhesive molecules present in the lymphoid niche, but gelatin can only present one type of or vitronectin mimicking niche or one type of structure. So to overcome that, we started working with a more designer system, which is a maleimide functionalized polyethylene glycol. And here the maleimide group can interact with thiolated peptides of interest. So you can choose collagen mimicking or VCAM mimicking any of those thiolated peptides, functionalize to the maleimide arm, and then cross-link using dithiolated cross-linker. This is called the Michael type addition reaction, very well established in the field. And at physiological pH and temperature, you can form these stable hydrogel structures with the cells encapsulated inside those. We benchmarked it against other known hydrogel platforms that undergo similar reactions, such as PEG acrylate and PEG minosulfone. And our very interesting finding is that the maleimide supports the survival of these primary B cells much more, as well as their differentiation as compared to the minosulfone and acrylate, which could be attributed to a lot of factors, including some of the known minosulfone toxicity to the cells. With that said, there are many applications in which minosulfone and acrylates work exceptionally well, but for all these mouse B cells and human B cells that we have tested, it seems that the minosulfone and acrylates are not really supportive of their long-term survival and differentiation. So with that, we could now functionalize with collagen mimicking peptides or fibronectin mimicking peptide or VCAM1 mimicking peptides, the sequences are shown over here, and start asking questions, the impact of the extracellular matrix, impact of various adhesive proteins, impact of time. And as you can see, in these conditions, for example, the VCAM compared to the VCAM1, which is this VCAM1 is presented by follicular dendritic cells in the B cell follicle. So you have a pretty stable epigenetic hallmark, but compared to a collagen mimic, which allows for a rapid drop by day 6. And the goal over here is to not have something go up and stay there, but it's more like a bell curve where the naive cells don't express much, then they go to the peak, but they should start exiting so that they can become plasma cells, memory cells, and therefore you need to have a better temporal control of their expression. So the designer system is allowing for that approach. We get asked many times, have you tested for antigen specificity? And now we have tested that in many platforms, including the human platform, which is in development. But here's a very interesting study that we did a couple of years back, where we took a transgenic mouse called B18. This mouse makes antigen-specific response to a small molecule hapten called NP or nitrophenol. And so we took B cells from here and put them in the organoid platform and took an approach to see, can we enforce selective enrichment of antigen-specific cells in an organoid platform? This is a very crucial question, which I can explain a little bit more later on if needed. But the key point I want to make here is that by using the antigen and using a T-cell-like signal, we're able to enrich for the cells that are antigen specific in the organoids while depleting the yellow bar, which is the antigen non-specific, which are the non-specific cells with that are still germinal center cells, but they have not been they have not acquired the antigen specificity. And further validated this using another model in which you are using PE, which is phycoerythrin based system, and we can show that by using specific combination of T cells and the stromal cells, we're able to generate a significantly higher antigen specific cells just by taking wild type cells. Here is a transgenic engineered mouse. Here we're using wild type system where we're still able to get PE specific response in this. So the next couple of slides focus on the application of this as well beyond fundamental immunology. One of those is in the area of predicting B cell immunity in aging against bacterial antigens. Many of you are aware that the antibiotic resistant bacteria they pose a major global health crisis. There are not really any vaccines out there to tackle those. One of those is the Klebsiella pneumonia, which actually fails to induce in vivo germinal center response or antibody responses. And so developing antibody immunotherapies ex vivo and then infusing back into the patient could be a promising alternative. But what we don't know is how to proceed from beyond this idea. What it means is, what kind of, for example, components of the bacteria should you take so that you can induce a better germinal center response, those kind of things. To address this, we collaborated with Dr. Ilana Brito at Cornell University to take clinical isolates of Klebsiella and then purify outer membrane protein out of that. So it's a combination of various components of the outer membrane of this Klebsiella, or use purified soluble AMP protein as a comparative benchmark, and use that to study some of the key features of the germinal centers that are there, for example, histone trimethylation. And the idea was to compare, do you need soluble protein, or can you use an outer membrane cocktail over there, which will have a lot of various components from the bacterial membrane. And there seems to be significant difference between whether in terms of the histone trimethylation and various other germinal center markers, depending on whether you use the full outer membrane versus a soluble purified protein. So it allows you to select for some of those. Now, can you use this to predict immune response, or at least that histone trimethylation in aged B cells? because aged patients in the get exposed to this infection quite a bit in the ICU and other places. So for that, we used 2-year plus aged mice and performed the same comparative experiment. We actually did not have any idea whether we can even form germinal centers out of these cells, because there is a known literature of immune B cell senescence, as well as defects in the T cell maturation process, which leads to poor immune response in aged individual. But when you present the CD40 ligand, the idea is that in vivo you cannot do this, but ex vivo you can forcibly present the CD40 ligand, giving an opportunity to these aged B cells to undergo the germinal center reaction. When you do that, you see a nice germinal center formation over here, which is less compared to a young mice, but not surprising because the B cells are also aged over here, so they have limited capacity. But we can successfully make aged B cell germinal center B cells outside in a hydrogel-based platform. And when you look at your histone trimethylation, you see similar patterns over there. Aged cells have significantly lower trimethylation in the germinal center B cells. And when you use Klebsiella outer membrane versus soluble OMPE, you can predict which component is going to lead to more trimethylation and try to circle back and come up with the therapy that might help in the improving the B cell antibody responses in aged individuals. So this is a project ongoing in the lab. We have some exciting new work that should be coming out in some time in this space. One of the things that I learned in Rinty's lab was working with the lipid nanoparticles, so the liposomes. And we've taken that approach more recently after many years of gap in working with liposome, to come back and ask a very interesting question, not as a delivery system, but more from a fragment of stromal cells. So in vivo, follicular dendritic cells present antigen to the B cells. So these B cells snatch these antigen, as shown over here, and then undergo reaction. When we present antigens as soluble or solid bead-based presentation or immobilized surfaces, we don't have that membrane fluidity that you have in a follicular dendritic cell membrane. So to mimic that, we have been using these lipid-based particles as fragments, where you can bind the antigen, functionalize the antigen to the surface, and then allow the cells to extract that from that surface upon binding of the B cell B cell receptor. And then we determine whether there is an impact on the germinal center process. A very simplified data is shown over here for these about 100 nanometer size liposomes, which shows that when you use soluble particles, soluble antigen versus liposome, you have a significant increase in the proliferative behavior of these B cells, as well as downstream germinal center phenotypes that we have. This paper should be coming out in next couple of weeks, suggesting how you can now go back and take some of these particle-based approaches and make them more like fragments of the cells, instead of using them as a delivery vehicle for a variety of applications over there. So we're excited about this. I think this will allow us to create these mimics of the follicular dendritic cells, which we have lacked in the field. Not the whole cell, but fragments of those, and hopefully this will allow us to better understand the B cell maturation process there. Okay. An ongoing work in the lab is we've transitioned since last year with support from Welcome Leap HOPE program to really take all of the work that I showed at the mouse level into the human system. So currently leading this project, we're building immune follicle on chip by human tonsils and PBMC derived blood cells derived response, so that you can predict infection and vaccination responses, adjuvant responses, by, for example, creating these human tonsils or human lymph node like structures, Then we're integrating that with the gut system with Dr. Andres Garcia at Georgia Tech, so creating mucosal immunity interface, and also lymphoid lung interface with Dr. Krishnendu Roy at Georgia Tech to understand the systemic immunity and the mucosal immunity. So this is an exciting project. I want to show how the human tonsil looks like. This is a spatial omics we have taken. So here's collagen in the human tonsils. These are the basal follicles and germinal centers that are being formed over here. And then when we take freshly isolated cells from tonsil donors, put them in a similar biomaterial-based platform, expose it to the human influenza antigen, and we can see that there is a much more significantly higher induction of germinal center response, formation of dark zone and light zones through confocal microscopy, and that we have now mapped this for temporal response over many days to see how you can predict immune response in a dish for a human system across multiple donors across time frames. So it's a very exciting project that we're also recruiting postdoctoral fellows on this. So if any of you are interested, please email to me, and hopefully we hope to submit this work in near future for publication. Over the next couple of minutes, I want to transition to my second part, which is development of these nanowires or nanoneedles to pre-program T-cell trajectory using microRNA. This is work that is now nearly complete from our side. For the first part, we're using microRNA as a rheostat for CD8 T-cell differentiation. And one of our collaborators, Dr. Brian Rudd at Cornell, has published some very important work that shows that microRNAs are critical regulators of cell fate in various T cells, including CD8 positive T cells. And there are several of these. So for example, Me29 is there, Me130 is there, and they function as a rheostat for these cells. So for example, a Me29 upregulation can take a naive T cell into a memory cell. Downregulation of Me29 and upregulation of 130 can switch it into an effector T cells. So our goal over here was to develop a technology where we can deliver these microRNA to these T cells in a very effective way. So historically, they have been these genetic materials have been delivered using various lentiviral-like approaches. There are some elegant cell squeezing technologies out there as well. But our goal was to develop a technology that will deliver these in a naive state without activating the T cells. That's a major drawback in the field, that many of the existing techniques can either kill the cells or activate them during the process of transfer of the genetic material. So we used a silicon-based etching-based approach to really create these tiny needles that you see, which are about 10 nanometer tip. And when you put the T cells on those, a typical T cell can occupy about 6 of these posts, and these needles poke very gently into these T cells. And the inspiration from this actually came from Dr. Stevens' work that she showed earlier, with the microneedle work penetrating, and the nanoneedle work that she showed. So the key point over here is that by using this technique and functionalizing the surface of these needles with cationic polymers such as polyethylenimine, covalently conjugating, you can dramatically reduce the toxicity effect of PEI and deliver genetic material through this. So for example, if you take a two-dimensional system with microRNA 29, you have high viability of the cells, but barely any delivery efficacy over there. When you take microRNA, when you take a nanowires that are functionalized with PEI, covalently conjugated, and you deliver either inert or Me29, you have a very high efficacy. Now shown over here is physically adsorbed PEI. There also you get high delivery efficacy, but you get poor viability because the free PEI is toxic to the cells. So with this approach, we have been delivering Me29 and evaluating downstream transcription factors that function as a rheostat over here. And then, as you can see with the microRNA 29 and nanowires, you are able to reduce the expression of specific transcription factors. For example, EOMS is responsible for determining the effector cell fate. So you can reduce that. You can reduce the various cytokines, TNF alpha, interferons with this. The next question we asked over here was, can you deliver more than one? And I want to make a key point over here is that the delivery is not the only thing that we're looking for. We're also looking for, are we activating the cells later on at our own wish by first pre-programming, or are we just during the delivery process, we are activating them? So we can deliver multiple microRNAs without activating the T cells and then go and add factors that will activate them. So we have a full control over their trajectory and pre-programming their trajectory right from the beginning. So that's what we are able to do over here by delivering more than one microRNA to the same T cell and regulating its expression of cytokines and transcription factors. We've now validated in multiple infectious disease models. So for example, there is a flu model that we have used that shows excellent response with these dual delivery of microRNAs as compared to the comparative groups. We've also tested with a listeria model and some cancer therapy models with this. So my last slide over here is going back to the inspiration. I've learned my career started in the biomedical engineering. I was a biochemical engineer before that, very interested in joining a distillery before I joined Rinty's lab and became a biomedical engineer. She inspired me to work in the area of nanoparticles for various applications, and I've continued to do that. I didn't get to show a lot of this work in the area of nano vaccines and nanoparticles that we have done, but if you're interested, please go and read some of these articles. And I want to thank my group for all the work they have done. I've been very fortunate to have these outstanding students, postdocs and PhDs, masters and undergraduate students throughout my career, and wonderful collaborators that are highlighted over here. The work that I showed, some of that over here in yellow, and my team and definitely my funding agencies, without which this work was not possible for us. Thank you.
Thank you very much. Thank you very much, sir, for the session. Before we move on to the next session, we would like to have a simple question, one simple question. Is there any questions? Is there any questions?
Yes, there is a one question. What now? Take a second.
There is one question.
There is a one question which is asked by Yogini. Is the technique used for treating autoimmune diseases?
Which one? The organoid platform or the nanowires?
Yogini haven't mentioned. I guess it's the first one.
The first one, yes. So, so we are investigating some aspects of that because the B cells are central to the autoimmunity in lupus, for example. And so we have started investigating some aspects of that to see how we can model some of those immunity. And yes, the T cell aspects can be expanded in that direction as well. We haven't worked on that yet. Yeah.
Professor, your quick comment on the other technique, the nanowire technique. She said it's a both. She was asking for a both.
Yeah, so the nanowire technique of yes, you can develop these T cells that can be used for targeting various autoimmune disorders, and you can pre-program them, you can expose them to various conditions and prepare them. Theoretically, yes, we haven't tested those. And for the organoid, yes, you can. We're already studying some of the lupus-based model in that.
Yogini, can you please ask your question quickly because we are actually running out of
Yes,
time.
thank you, sir. Thank you, sir, for this wonderful explanation of this new technology. I want to ask another question. In one of your experiment, you have mentioned that with this technique, we can change the fate of the cells from memory to effector cells. So, is this study can we use in treating allergies?
Yes, you could potentially use that. You will have to define the specific microRNAs, how you want to use those, and which subtype of T cell that you want to use. But yes, by selecting those, you could change the trajectory and apply to some of those applications. Yes.
Okay. If audience wanted to ask any question, just put in the chat box. We'll ask Professor Ankur later, and then we'll take his response and return it to you. We'll limit to you definitely. Thank you, Dr. Ankur, for your wonderful talk. Actually, there's a lot to learn from your today's talk. So we'll definitely email you if you have any questions. Sure. Thank you. Have a good day. Next, we'll have a next lecture.
Ladies and gentlemen, please welcome Professor Nicholas A. Kotov from University of Michigan. Professor Nicholas is a Joseph B. and Florence V. Setska Professor of Chemical Engineering at the University of Michigan. Professor Kotov pursued a PhD from Moscow State University in 1990. His postdoctoral studies encompassed the synthesis and self-assembly of biomimetic nanocomposites. Today, his group uses this expertise in producing and manipulating chemicals to energy, medicine, electronics, and advanced materials with new properties. Professor Kotov serves as an associate editor for the journal ACS Nano and as an advisory board member for several other nanotechnology journals. He has received awards and recognitions, which include the 2017 Colloid Chemistry Award of the American Chemical Society, the 2016 Stephanie Kolick Award of the Royal Society of Chemistry, the 2016 UNESCO Medal for Development of Nanoscience and Nanotechnologies, the 2014 MRS Medal, and the 2013 Langmuir Lecturer Award of the American Chemical Society, and the 2012 Stein Award from the American Institute of Chemical Engineers. Professor Kotov is also the co-founder of 5 startup companies and a passionate advocate for scientists with disabilities. With these words, let us welcome Professor Nicholas A. Kotov from University of Michigan.
Prof. Kotov.
Thank you very much, Mr. Chairman. It's a great pleasure to be part of this meeting and the great honor as well. So let me start sharing my screen, and let me start by acknowledging the organizers for the excellent job that they have done with this meeting. Just a second, let me figure out how to click. All right, so I think it's on right now.
Yes.
Okay. Wonderful. I'd like to talk to you today about the big field which is emerging now in translational medicine and the nanoparticles in particularly. These are the chiral nanoscale structures, and they encompass in them a lot of possibilities for a number of biomedical applications. And in this talk, I'll go over a few of them. First, I would like to talk about some fundamentals of that field and why the chiral nanostructures have a lot of very strong optical activity. Then I will address some aspects of that which are related to engineering of biomedical structures and biological applications, and this is related to complexity. And I would like to talk about how do we approach assessment of biological complexity and translation of that into the nanoscale structures. And in continuation of the previous speakers, I will talk about some applications in the space of immunology and drug delivery. At first, I'd like to talk about the memory of Professor Hrinti Banerjee, and some of her words really inspired me, and that is the inclusive healthcare. And she worked a lot on the nanoscale structures and nanoparticles and liposomes. And I'd like to continue that topic in this presentation, extending it not just from the empirical optimization of nanoscale structures to the true numerical mathematics-based engineering of these materials. And one of her works that contributed directly to this field include the works on the nanovaccines and gold particles. First, I'd like to give you why so many people are interested now in chiral nanometer scale structures. And primarily, this is because they have incredibly high polarization rotation. I will talk about other aspects as well, in particularly in respect to the biomimetic applications. But now let's address the fundamentals here and why, say, the nanoparticles from gold or semiconductor or some ceramic materials have very strong rotatory strength. And polarization rotation or circular dichroism is proportional to the imaginary part of transient electric and transient magnetic dipoles. And if we just look at the one of those, the other one is very similar. The transient electric dipole very strongly dependent on the type of the material these particles are made of. It's proportional to the square root of dynamic polarizability, which changes dramatically from organic molecules to semiconductors and to metals. And as you can see here, 7 orders of magnitude increase in dynamic polarizability accompanies the transition from organic matter to the metals. And of course, then the transient electric moment will increase 3.5 orders of magnitude, and indeed it will be very strong polarization rotation, which for the first time was observed in nano-assemblies of gold for the plasmonic frequency of this metal. So it is in near infrared part, which is very convenient for a number of applications in biological space. And this work was done in collaboration with Professor Chu from Wuxi University. And with him and others, we explored the utilization of this incredibly high optical activity for biological sensing, for example, for detection of cancer and other health problems. And we found initially that we can extend the sensitivity of these methods by using PCR and similar techniques to the attomolar detection of long strands, very informative strands of DNA, and then even to zeptomolar. The reason for such high selectivity and sensitivity is because the physics here is very advantageous, but also because in that plasmonic part of the spectrum for gold, there are not so many organic molecules which have this similar polarization rotation. And that's why it's actually very universal and very informative. It's also quite different from the absorption because in absorption, the traditional red-blue arrays, they reveal much lower change in optical activity, especially when a DNA strand is large. It's now undergoing the translational efforts for sepsis, as well as we're working on the detection of viruses. But now let me talk about some of the new parts of this work, which is related to high complexity. It may sound abstract, but it's actually very much related to what Professor Banerjee and others refer to the democratization of the healthcare, accessible one. And if we understand the mathematics, if we understand the engineering of the structures, it will be available for every scientist around the world and every practitioner. Why do we need the complex structures? Quite simple, because the biological structures we're trying to fix and replicate are complex. So, for example, if we take bone, it's made by hierarchical self-organization of nanoscale pieces of inorganic matter integrated with the biological factors. And because of that hierarchical structure, which involves the chirality as well, it is able to have such a wonderful set of properties which enables us to combine the strength and toughness and lightness in our bones. And it's hard to replicate it by any other material if it doesn't have that. Of course, there are some other applications, for instance, in replication of light sensitive membranes, which also are organized by self-organization and have a number of properties which are related to the complexity of the structure. So now let's look at our building blocks, and this will be the nanoparticles. So how do we use the chirality and other properties of nanoparticles to build, to engineer these complex biomimetic materials or something like that? There are a number of problems here, fundamental ones, and one of them is that the nanoparticles are often dispersed. So even the best examples of monodispersed, quote unquote, nanoparticles actually have a variety of differences that are slightly different in size, slightly different in organization of the surface layers. And they're certainly much more diverse in their structure than the biological counterparts, for instance, the peptide chains, which are engineered with the molecular precision. The second problem is the thermodynamics. And we have to admit that if we form the complex structures, which are in the level of high energy state of this landscape, so if we imagine the system of the nanoparticles which self-assemble, they tend to go to the high mass states because that state enables one to maximize the attractive forces, for instance, van der Waals attraction. And that means that we need to control that system and to prevent it from reaching these high mass states. How do we do it? We have to impose some restrictions on its organization, and in the past, in collaboration with Professor Sharon Glotzer from the University of Michigan, we discovered that electrostatic repulsion works as very good restriction. And it's actually very typical for liposomes, for micelles, for a number of protein assemblies. For nanoparticles, it's easy to observe because they can be investigated by electron microscopy, and we saw quite uniform assemblies of almost spherical supraparticles, which made from several hundred of nanoparticles. And the reason why they are so uniform is that at some point, the nanoparticles which might want to join the supraparticles and have some attractive interaction, they can't because of this long-range electrostatic repulsion. And this enables us to control the evolution of this system from reaching these high mass states. And let's try to remember that and try to address the problem with the polydispersity of the nanoparticles. So what if we make the particles chiral and polydisperse in the same time? And we tested this approach for the nanoplatelets of gold sulfide, which are coated with cysteine amino acid on top and the bottom of these nanosheets. And certainly, as you can see, these nanosheets are very polydispersed. And after that, we decided to figure out how we can assemble them and whether the chirality makes a difference in what structures we're forming. It does. For example, if we use the top and the bottom layers from L-cysteine, we form quite uniform and good-looking structures, which we refer as chiral hedgehogs. If we change L to D, enantiomer of cysteine, we also form very similar structures. But if we look at a racemic mixture of the cysteine, we form very different, although quite uniform too, structures which can be referred as, for instance, kayak particles. What's the difference here between L and D? It comes out when we look at the spikes of these particles. So each spike is actually a stack of the nanosheets of gold cysteinate, and these nanosheets are twisted and fanned, and the direction of each of that twisting and fanning is opposite for LND enantiomers of cysteine. This translates into a number of interesting properties, but one piece which you might appreciate is that electrostatic restriction, it's not just a methodology for making good science, it's also a methodology for translation. And as you can see, we can make these particles in big batches with high dispersity, despite the fact that original particles are actually very poorly dispersed. Electrostatic restriction enables us to scale it up quite easily. Also, the chirality enables us to form a variety of different particles. So this is just one example of a diagram that is possible for these self-assembled complex systems. You can have here some superparticles from nanosheets, or twisted bundles from nanoribbons, or these asymmetric chiral stars, and maybe supraparticles from supraparticles. The hedgehogs themselves are in top, bottom, left, right side of these diagrams, and kayak particles are in here. So, but back to science. So, which of these particles are actually more complex? It is not just an abstract problem because it enables us to compare it to the biological structures and also answer the fundamental engineering questions. So how do we approach it? In order to answer this question, we developed the graph theory approach to evaluation of nanostructures. We describe the nanostructures as a graph, which is a set of nodes and edges. It's fairly simple, and the complexity is a big part of the graph theory, and it's also used in a variety of other fields, in humanities, in economics, in social science, the networks, and in order to measure the complexity, we can apply different set of mathematical values. For instance, it could be multifractal indices or connectivity index, so how connected the structures, how complex, how many edges each node has, or the complexity index. We used in this work the complexity index, which we developed based on the works of Randich and Plavich for the chemical structures. And we extended it to the nanometer scale assemblies, to semi-infinite structures, and to the structures which are non-atomic and based on nodes which describe the nanoparticles. This approach is applicable to particle chains, to particle rods, to the nacre-like structures which are used in bone implants, or some other applications for graphene, supraparticles, and superlattices. So how do we do this? Actually, it's not very difficult. So if we have a nanoparticle, it may be spherical, it may be random shaped, but fairly isotropic. We denote it as a node. If the particles are connected through, for instance, organic phase, as they commonly do, this connection is described as an edge. But particles, unlike atoms, can have a variety of shapes, which make them more complex. And for instance, if these rods, we will use the minimal representation of these one-dimensional rod-like structures as K2 graphs, which define a line. The same is true for nanosheets. They're defined by a triangle. The chiral nanostructures, or chiral structures period, they have the minimal representation as K5 graph, which denotes that in the three-dimensional world, this is the first graph which have to be extended in 3D, and the chirality is in 3D. The rules for connecting them are still the same. So if there are some connection through the organic phase or through some other different phase, this will be denoted by the edges. So now this math needs to be checked, and let me give you some idea how to translate our expectations about the complexity and how they match or not the data for complexity of the nanoscale structures we engineered. So this is the formula for complexity index. It's a limit. It's infinite sum of slowly decreasing values. And for, say, nanoparticle pair, it will be 1.5. It's more complex than individual particle. Simple enough. If it is a chiral structure, for instance, a twisted ribbon, the complexity index of K5 graph is 12, which is higher than this. Also makes sense. If it is a chain, which are actually quite common in biology and nanostructures, these are described by the repeating sequence, so it has to be more complex than, say, a pair, but eventually information content here is not much more difficult, not much more different than the information content in a pair. It's higher, but not by a lot. And that means indeed that the complexity index makes sense, which is 6. Higher than in pair, smaller than in the twisted ribbon. Now we can apply the same technique for the evaluation of more complex structures. For instance, these supraparticles, which are made from nanosheets. So supraparticles determine that they're all about the same size, they have the phase boundary, and the building blocks are sheets. The nanosheets are also involved in nacre-like structures, in bone-like structures, and in this case, these are nanosheets which are connected through the organic phase, the proteins, and they are semi-infinite. The complexity index will be 24. And here I would like to point out an interesting observation. So the higher the complexity index, the more useful this structure is. And this may not be that obvious, but I'll give you an example that these kind of structures are used by all biological organisms. And it can be used in bones, and can be used in shells, it can be used in some other organs and tissues, but this is certainly very well utilized structures. Now let's have a look at the questions about this phase diagram and whether, say, kayak particles are more complex or not than the hedgehogs. We can calculate the complexity index based on the same rules and electron microscopies. These are not the structures or these graphs are very much rigidly bound to what we observe at the nanometer scale in these materials, in these particles. We have the complexity index 40, and here 87. So the difference comes from these chiral sheets, which are twisted and fanned. And that's why the complexity index is high. And that's why, perhaps, and you will see it, that the chirality is very significant for biology because it enables one to reach high complexity. And homochirality is something which is important because it translates through scales, and it enables us to exist as a very sophisticated, complex interacting structures. With this thought, we might also want to compare not just the particles that we made in the lab, but also the biological particles, which are very similar. In fact, they were the prototypes of these particles, and these are the vocalists. Actually, their complexity index is smaller. And it's not surprising eventually, because once we learned how the structure is organized in the lab, we can make more and more complex, more sophisticated structures. Now, I'd like to point out that particles are used in a very wide range of drug delivery applications. And they need to be porous, they may have different functionality. For instance, these particles have gold, and engineering of this sensing and drug delivery and multifunctional particles will require higher complexity. And now we not only can compare which particles are more complex, which is not, but also can draw their structures and engineer, just like in chemistry, based on, for instance, the functionalities which are associated, for instance, with spikes or porosity or multi-shell organization, which needs to be for the drug release. Now I'd like to address the question about how the complexity is useful. And this comes through these studies of biomimetic nanostructures. And I'll give you two examples. One of them is based on individual nanoparticles. Actually, both of them are based on individual nanoparticles, forms complexes with self-organized biological structures. So there are some works which are emerged in the literature extensively about the use of inorganic nanoparticles as biomimetic substitutes. And one work that I like to highlight from the group in Turkey and Spain is that the carbon graphene oxide particles can reduce the activity of SARS virus. And now the question is, how do we engineer these particles? In the past, in our work, we did it actually empirically and used the different shapes of the particles and looked at how does individual shape changes, for instance, antibacterial activity and association with beta-galactosidase, which is a key metabolic protein of a lot of bacteria. And we saw that the pyramidal shaped zinc oxide particles strongly make a complex with beta-galactosidase and can inhibit even very aggressive antibiotic-resistant bacteria. So this is a different approach than traditional antibiotics. And that started our work, which will be in progress out very soon. How do we engineer the nanoparticles based on our knowledge of the protein-protein complex? So in the previous examples that you saw, these are the nanoparticles which replicated another protein, forming a complex either with spike of SARS virus or with beta-galactosidase. So we started learning about protein-protein complexes and have realized that if we just use the geometry of these complexes, we can potentially predict how the nanoparticles with similar geometry, similar chemistry, can interact with the same proteins. The basis of our description of protein-protein complexes was by using the graph theory exactly that topic that you just have seen, and understanding how we can describe these very complex, sophisticated structures by the graphs. And we used the graph theory to impart descriptors to the proteins and then translate it to the nanoparticles. So we used three types of descriptors. One is traditional chemical descriptors, which are associated with localities and with amino acids in the proteins. The other one is geometrical descriptors, which are based on concavity of the structures. And the other one, big sections, is the graph theory descriptors, which evaluate the protein and particle as a graph and take out the Ricci curvature, the multifractality index, which I referred to, and the Gaussian network. Among all of those, we also use the chirality index as a very fundamental descriptor of the geometry of the structures. And so I can give you some examples of how say the pocket geometry changes, and it's very variable. So if we look at the structure of the protein, there are many descriptors, many areas which are capable of distinguishing themselves with other proteins. If we look at the graph theory network, the same is true. If we look, for instance, the Ricci curvature, or how one part is connected to the other part, is basically a measure of the transport in the network. For the proteins, it's a measure of their reconfigurability. It also very variable. And so you see here very stiff part and very flexible part. Fractal dimensions are also quite interesting. So these are rules of the network of the graph. And here you can also see how variable it is. And I would like to highlight that variability as a distinction between one molecule and the other. Chirality is rarely used in drug discovery, and we implement it here for the protein-protein complexes, and we implement it locally. And so you can also see how it can be different. It's actually can be left and right. It's not just about amino acids. And chemical features are very well known. So how do we do it? So there are many descriptors. We pack them in these geometrical matrix of distances that you saw before. And after that, each protein has a matrix, each protein complex has a matrix, and we analyzed them. Of course, we used the machine learning, and these are the algorithms that we applied. And one very striking finding here that we observed, that if we use just the graph theory and geometry, we actually have a very accurate description of the predicted complexes in terms of yes or no, form or not, and the locale, so how, what are the groups which are involved. Chemical components, that is, say, hydrophobicity, charge of amino acids, are not very influential here. And we can discuss it later why, but the point is for nanoparticles is that now we can extract the chemistry and use just the geometry, and indeed, just by using the graph theory and the geometrical features, including the chirality, we can accurately predict a number of protein-protein complexes, even those which were not included in the database. And we can predict the reaction sites, the interface sites, as well as the affinity. And we can do it for a number of protein-protein complexes. And now the nanoparticles. So why this is important? Because by using this universal descriptor set, which is applicable both to the proteins and the nanoparticles, we can now translate the knowledge of the protein-proteins interactions to the nanoparticle protein interactions and engineer, therefore, the structure of the particles. And we tested whether it's true or not, and we saw that the predictions based on our algorithm, how, say, the graphene nanoparticles, hydroxylated graphene, from this study, which used the molecular dynamics of Reedy et al., predicts exactly the same thing as the extensive molecular dynamics studies. Another work from the biomedical field, which is quite extensive and quite puzzling, is the existence of protein corona around the particles. And it's very difficult to say which protein will be there and which protein is not based on a priori assemblies, a priori knowledge. And that work by Rebecca Pinals and Marquita Landry and others, they show that these are the proteins which are abundant in the that shell. And our methods exactly predict the same thing. So with that, I'd like to point out that this is the tool which now becomes available. And if you have any questions, I'd like to, I'll be happy to share it. Now, some other interesting study which shows how the complexity of shapes make a difference in the biological applications in biomedicine. So these are the chiral nanoparticles of gold, and it's also the collaboration with André de Moura from the University of São Carlos in Brazil and Professor Chun Lai Shu from Jiangnan University in China. So these chiral nanoscale particles are complex, and they are also uniform, and we can not only vary their shape, but also vary their chirality. So we now have the tool to tune the extent of chirality by illuminating with chiral circularly polarized light. And the question that we're asking here, if we change the chirality, how do the changes with biological response vary? And we can measure the chirality by optical methods, by, for instance, G-factor or circular dichroism, or by the same chirality index that we saw for the protein OPD. So indeed, we can vary the chirality of particles here. And what we decided to choose as a testing bed is a very complex system of immune response starting from bone marrow dendritic cells. In here, the role of the particles is coadjuvants, and they're basically sending the activation message to the dendritic cells, start learning what antigens are. And the reason why we thought that chirality here will be important is because there are a number of molecules on the membrane of dendritic cells which have the nanoscale dimensions and they're chiral. These are the receptors CD97 and the MR1, and they have long sugar tails which are wrapping around like springs around the particles. And so what we looked at, all the elements of this sequence of events and eventually to antibodies. So what we learned is that if we use just a standard of albumin for activation of the cells, then we see that chirality matters. So that complexity of the particle shapes, that twisted nature really makes a difference here when the cells are interacting with chiral structures in biology. And as we increase the chirality, the immune response is also increasing. L type of chirality is causing the larger immune response than the D type because it interacts strongly with CD97 and EMR1. And the same is observed in another test. This work started about 5 years ago. And of course, now we would have used COVID virus, but at that time we used H9 and 2 influenza antigen. The same thing is observed here. L type of chirality interacts strongly with the sugary tails, and it induces stronger immune response than the D type. And with that, I'd like to offer you this as an example how now we have the toolbox, the conceptual foundations, how to use the chirality and complexity for the biomedical applications. These are the wonderful folks who made this possible. These are also wonderful folks who rarely get acknowledged in person, but should have been, who funded this, and I'd like to thank you for the wonderful opportunity. And I'll be happy, of course, to answer any questions.
Yeah. Thank you, Professor, for your wonderful talk. We'll like to take only one single question because we are running again a bit short of time.
I understand, yeah.
So any quick question would be appreciated. No. Okay. I have a question. So you mostly spoke about the chirality, right? Have you considered about the surface charge of the different shaped particle which you develop by using chirality for the cellular interaction, especially for the immunization?
Oh yes, absolutely. Charge is a factor which is often used. And we decided actually to, and we certainly incorporated charge in the descriptors of amino acids and particles.
Right.
Charge is a kind of a tricky thing, as you know. I mean, if you want to increase interaction and make the particles positively charged, you also increase increase the toxicity.
Right.
So that's a disadvantage. So we thought that we need to use a different methodology, and chirality had offered us that approach.
Right. Okay. Does anyone have any question, or shall we thank Professor? No. Okay. Thank you, Professor, for your
Thank you
wonderful
so
talk
much.
today.
It was a wonderful opportunity. Thank you.
Same here. Thank you, Raj.
Ladies Ladies and gentlemen,
Ladies and gentlemen, our next speaker for the day is Dr. Ladies and gentlemen, we are delighted to have with us Professor Darrell Irvine, who joins us from Massachusetts Institute of Technology. Professor Darrell J. Irvine holds a PhD in polymer science from MIT. After this, he continued his postdoctoral research in immunology at Stanford University. Professor Irvine joined MIT in 2002 as an assistant professor of biomedical engineering, Department of Material Science and Engineering, and then the Biological Engineering Division. Currently, he is a professor at MIT and an investigator of the Harvard Hughes Medical Institute. He also serves on the steering committee of the Rogan Institute of MGH, MIT, and Harvard. He aims to engineer immunity through a fusion of immunology with biotechnology and material chemistry, employing material science-centric approach to create new therapies based on the controlled modulation of immune system. Dr. Irvine's interdisciplinary work has been recognized in numerous awards, including a Beckman Young Investigator Award, an NSF Career Award, selection for Technology Review's TR35, elected as a Fellow of the Biomedical Engineering Society, and appointment as an investigator of the Harvard Hughes Medical Institute. Ladies and gentlemen, please join me in welcoming Professor Darrell Irvine.
Thank you.
Welcome, Professor. Would you like to share your screen?
Yeah, so I'm trying to do that. Okay, can you see this slides okay?
Yes.
Okay, well, thank you so much to the organizers for inviting me to take part in this memorial symposium. I'm very honored to be here to help celebrate Professor Banerjee. It's been inspiring to hear the words of her impact on her trainees and colleagues. I'm going to share a few connected stories from the lab from our recent work. A lot of the effort in my laboratory is focused on drug delivery targeted at the immune system. We work on vaccines and cancer immunotherapy. And a lot of what we like to do, you could think of as trying to control the timing, the dose, and the location of where we're delivering stimuli to immune cells to try and promote either vaccine-mediated immunity, T-cell responses, antibody responses, or anti-tumor immunity. And I'm going to tell you 3 stories that are all connected by a common technology platform that we started studying more than 10 years ago now, but which, as we've continued to study and understand these simple molecules I'm going to tell you about, they've continued to inspire new application ideas in the vaccine and immunotherapy space. And where the story starts is some work where we were trying to understand how to generate more potent vaccines for cancer. And we were particularly interested in a class of vaccines known as peptide vaccines. These are vaccines formulated by taking 20 or 30 amino acid linear peptide epitopes and combining them with adjuvants to inject and try and elicit T-cell responses. These peptides would be either derived from mutated regions of a protein expressed by a tumor or proteins uniquely expressed by a tumor, where you take a small chunk of that larger protein sequence and use it as an antigen. Peptide vaccines are very safe. They're inexpensive. Because of their simplicity, they're amenable to rapid GMP manufacturing, and that has allowed, for example, the advent so-called neoantigen-based vaccines, where a patient's tumor is sequenced and peptides are identified that represent mutations specific to an individual person's tumor. And if you look at the clinical trial frequency over time, you can see there's a steady increase in peptide vaccines over the last 10 or 15 years. But a major limitation has simply been in general, a low potency of these vaccines for priming CD8 T-cell responses in humans. And where we started was an interest in trying to understand what controls the potency of these vaccines. And because of our engineering background, we approached this as a sort of physical chemistry problem. And we started with a very simple question: do these vaccines get where they need to go when we inject them into the body? So vaccines are typically injected into the skin or into the muscle, but all the action needs to happen in the downstream draining lymph nodes. So those antigens and adjuvant compounds we inject need to be transported through draining lymphatic vessels to reach the lymph node, where they can be taken up by dendritic cells and processed and presented to T cells. And in some very basic initial experiments where we fluorescently labeled peptide antigens, injected them into mice, and then took the draining lymph nodes out at different time points after injection, we found unexpectedly that very little of the vaccine dose reached the draining lymph nodes. The signal was barely different from an uninjected control. It was compared to a positive control where we injected these vaccines directly into lymph nodes ex vivo. Here we're looking at whole lymph nodes by whole tissue fluorescence imaging. We could see that something like less than 0.1% of the dose of the vaccine was actually reaching these critical tissues where the immune response is initiated. And as we started to study what causes this outcome, we unveiled the following sort of simple picture, which is that on injection into tissues, there are two main exits for injected drugs. They can either enter into the blood vasculature or clear into the lymph. And to first order, the fate of injected compounds is governed by molecular size, plays a huge role. If you're small enough to diffuse between the basement membrane and tight junctions lining the blood vasculature, because the blood clears tenfold more fluid per unit time than the lymph does, then if you're a small enough compound, you'll go into the blood, whereas larger macromolecules and particles tend instead to traffic through the tissue and enter lymph vessels, which is where we want our vaccine to go. And this has been quantified by other groups in large animal models for series of globular proteins, and showing if you give a subcutaneous injection of a series of proteins of increasing molecular weight, which here is just a proxy for molecular size, you see this very clear relationship where the bigger the molecule, the more of it on injection will appear in the lymph. And this data clearly explains our peptide vaccines, because we're injecting these a couple kilodalton peptides down here, and that is predicted to go about 98% directly into the bloodstream, where it will be diluted and degraded. And this is also where low molecular weight adjuvants are going to be trafficked, and this leads to not only lack of efficacy, but tends to increase toxicity from systemic exposure. But you also see that in this relationship, at a threshold of about 40 or 50 kilodaltons in globular proteins, corresponding to a hydrodynamic radius of 3 or 4 nanometers, there's this plateau where the vast majority of the injected dose is going into the lymph. And as we were thinking about this problem and how to address the limitations of peptide trafficking, we realized that the most common, the most prevalent protein in blood and interstitial fluid, albumin, is just the right size at about 67 kilodaltons, that it's constitutively moving from blood to lymph. And so that inspired the idea of looking at albumin as a molecular chaperone to carry our vaccines to lymph nodes. And to do this, we devised a strategy where we use one of the main biological functions of albumin, which is it serves as a fatty acid transporter. It has 7 different lipid binding pockets. And we started designing vaccines, which we call Amphiphile vaccines or AMP vaccines, where we take a hydrophobic phospholipid tail and link it to either molecular adjuvants like CpG DNA or peptide antigens. And in water, these molecules will form micelles, but if we select the phospholipid tail properly, in the presence of the high concentrations of albumin that are present in vivo, these micelles will be broken up, and these molecules will be associated as unimers with albumin molecules that would then traffic, we predicted, would follow the biodistribution of albumin and be carried out of the tissue into the lymph vessels. Now, we carried out a series of studies to sort of learn the design rules of these molecules, and I'll just summarize them in one slide. So first, we found that if the more saturated, the longer the diacyl tail of these molecules, the more hydrophobic they were, the better they bound to albumin, and the better they traffic to lymph nodes. So here's a series of lipid tails of increasing number of carbons in the lipid chain, and you see the longer the lipid tail, the better the albumin binding affinity we measured in vitro, the better the lymph node delivery we saw in vivo. For delivering peptide antigens, there was also a second component to this design, which is we include a polyethylene glycol, a hydrophilic polyethylene glycol spacer to ensure that the molecule is fully soluble, because you can imagine some antigens are themselves quite hydrophobic, and if we link them to this hydrophobic tail, then we just get an insoluble mess. But by introducing a PEG chain of sufficient length, and you can see here as we increase the number of ethylene glycol units in this PEG spacer, we get greater solubility of the amphiphile molecule, and that again leads to greater and greater uptake in the draining lymph nodes after we've given a subcutaneous injection in vivo. Once we've optimized these two aspects of the molecule, we can take molecules like CpG DNA or peptide antigens that in tissue sections showed almost no ability to accumulate in the draining lymph nodes and convert them with this amphiphile tail into molecules that can impressively traffic to the lymph node. And this had a dramatic impact on the potency of peptide vaccines in mouse models. So if we now take these optimized amphiphile vaccines and immunize here against a peptide from a melanoma model, we could take vaccines that administered twice into mice, and then 3 weeks later we read out antigen-specific T cells in the blood. You could see that a traditional peptide vaccine against this melanoma antigen GP100 elicited a few percent of the circulating CD8 T cells recognizing the antigen. If we gave that same antigen as an amphiphile form, now half of the circulating CD8s in the animal are reactive to that peptide. And this is a tremendous increase observed across groups of mice. And that in turn led to greatly enhanced anti-tumor activity. So in the melanoma model, if we gave a vaccination against an antigen called TRP2 combined with CpG as an adjuvant, in this aggressive B16 melanoma model, the vaccine had essentially no ability to slow the tumor down. But with this optimized amphiphile structure targeting the vaccine to the lymph node, now we could substantially slow tumor progression and extend animal survival. And so based on this promising work, this technology has now been licensed to a startup company that's now in the midst of a phase 1 clinical trial testing this approach to vaccinate against KRAS antigens in pancreatic and colon cancer. So that was where we started, trying to do a better job of activating natural T cells with a vaccine against cancer. But as we were working on this approach, we got the idea that this could maybe also be applied to boost another kind of immunotherapy, chimeric antigen receptor T cell therapy. This is an adoptive cell therapy currently in the clinic, and which has reached licensure for several different hematologic malignancies. CAR T-cell therapy is based on taking T cells out of the peripheral blood of a patient, transducing them with a viral vector to express a chimeric antigen receptor, or CAR. And the CAR is basically a hybrid of an antibody domain that's going to bind to some tumor antigen linked to signaling components from the T cell receptor and co-stimulatory receptors on T cells. And this Frankenstein molecule now allows any T cell to become an anti-tumor T cell. When the CAR binds to a tumor antigen, it activates this T cell to kill the target. And after transduction, these T cells are expanded in large numbers and reinfused into the patient. And that has led to dramatic regressions, as I mentioned, in hematologic malignancies. I'm showing you here data from one of the very first clinical trials at the University of Pennsylvania showing patients in acute lymphoblastic leukemia, where about 75% of patients have a complete response against their tumor with this CAR T-cell therapy. However, the big challenge in the field has been in moving this therapy out of leukemias and lymphomas and into the treatment of solid tumors. So this is a summary from a few years ago of a number of clinical trials of CAR T-cells targeting different antigens in solid tumors. And you can see here the gray bars are for patients who were treated who had only partial, who either had progressive disease or just stable disease, essentially no real tumor regressions. And you can see that the majority of patients treated so far in solid tumors, with the exception of this GD2 CAR in neuroblastoma, have really not been very effective. And so it's thought that one of the issues in solid tumors is the immunosuppressive microenvironment that develops. But we also wondered whether a limitation could be from the way that we expand these CAR T cells to infuse them into patients. So after the viral vector transduction, we expand them in vitro using antibodies that activate the T cell receptor and co-stimulatory receptors. And this is a very non-physiologic means of stimulation. It's very efficient, but it's very different from how T cells are naturally activated in the body. And we wondered whether we could make CAR T cells more effective against solid tumors if we could take this expansion step and move it into the patient and allow CAR T cells to be expanded in the natural native lymphoid microenvironment of lymph nodes by creating something that would act like a vaccine for CAR T cells. And the inspiration for this came from one of the other physical properties of these molecules. So as I said, as we continue to study these amphiphile molecules, we continually learned interesting new features of their biology. And so one of the other features, in addition to albumin binding, is that these amphiphile structures will also happily insert their lipid tails into cell membranes. And if we add these amph molecules onto cells in culture with no serum present, you can use them as a beautiful membrane stain. And we found that if we analyze more carefully the cell level distribution of these molecules in vivo, by, for example, taking an amphiphile made of just a fluorophore, FITC, if that's injected into mice, as I've shown you before, these molecules very efficiently accumulate in lymph nodes. Importantly, they only get into the draining lymph nodes, and they don't distribute into the systemic circulation. So there's no accumulation in distal tissues like the spleen or the liver or the heart. But when you look into the lymph nodes, what you find is as these molecules accumulate in the node, if you sort cells out of the lymph node that have taken up these amphiphiles and stain them with an antibody to detect FITC, you can find that there's a lot of this amphiphile FITC still displayed on the surfaces of these cells, even in vivo. So this membrane insertion behavior happens both in vitro in a cell culture dish and in the native lymph nodes in vivo. And when we looked by flow cytometry at what cells these amphiphile molecules are decorating in vivo, we found that they were in fact decorating macrophages and dendritic cells, but there was very little uptake in T cells and B cells. And a day after injection, about 30 or 40% of dendritic cells were coated by these molecules, but by 3 days, these amphiphiles were cleared off of the cell surfaces, likely due to ongoing endocytosis. And the reason we believe there's this compartmentalized uptake into macrophages and dendritic cells just has to do with the anatomy of the lymph node. When lymph comes into the lymph node, it enters this region called the subcapsular sinus that's a lining around the outside of the lymph node. And then lymph is carried down into the T cell and B cell areas through collagen conduits that are lined by primarily dendritic cells and macrophages. And macrophages also line the sinus. And so we think the reason these amphiphile molecules are coating these cells is they're simply the cells lining the entryways that as lymph comes into the lymph node. But this turns out to be, and this is thought to be a mechanism by which the lymph node anatomically positions dendritic cells in a favorable position to collect any molecules coming in from lymph, because their job is to capture and present antigens. So we're going to now exploit this feature to create a means of vaccine boosting CAR T cells. And we do it in the following way. So now we create an amphiphile where we link not just a peptide antigen for a natural T cell, but instead a peptide or small molecule or protein that's going to bind to our CAR T-cell receptor. On injection, this CAR ligand is going to bind to albumin. It's going to get trafficked to the lymph node. But when it reaches the lymph node, it will transfer off of albumin and into cell membranes. And again, as I showed you, preferentially, it'll be coating macrophages and dendritic cells. And now a CAR T cell that arrives in the lymph node will see its ligand displayed off the surface of the dendritic cell, and it will receive not only stimulation through its CAR, but also have the opportunity to be activated through all of the many co-stimulatory receptors and cytokines that dendritic cells naturally display and secrete in order to program optimal T cell activation. And this concept was pioneered in the lab by terrific postdoc Leon Ma, who's now starting his own lab at Penn. And I'm going to show you how he developed this idea. So we devised two different ways to interface our AMP vaccine with CAR T cells. And I'll just show you this on one slide. And we found that both of these approaches work sort of interchangeably. So the first approach is we have a CAR T cell where the CAR antibody domain binds to a tumor antigen, but it also recognizes some surrogate peptide ligand, which we call a peptide mimetope, that just acts as a replacement for the tumor antigen. The second approach is to make a CAR that has two binding domains, a bivalent CAR where one antibody domain binds to a tumor antigen and a second scFv binds to any molecule we want. And in this case, we chose the simplest possible molecule we could think of. And we went back to this fluorophore FITC because there are many high affinity antibody domains known that bind to FITC. And so here, our Amphiphile vaccine will just be an AMP FITC molecule, and it will stimulate through this arm, whereas the tumor cells will be recognized through the other arm of the CAR. So first, we explored this idea in vitro. We took target cells and coated them with AMP FITC in cell culture. And you can see that as we increase the concentration of FITC in solution, you got steadily increased labeling of these target cells by flow cytometry. And if you put them in the microscope, again, you could stain with an anti-FITC antibody and show that even though there's constitutive endocytosis happening, for at least 24 to 48 hours, there's a substantial amount of this FITC molecule displayed on the cell surface. And now if we mix these labeled target cells with FITC-specific CAR T cells, those T cells would get activated, they'd secrete cytokines, they'd proliferate, and they would kill the target cells. So it looks like display of this artificial ligand off the target cell has all of the elements needed to activate the CAR T cell. So then we moved in vivo into mouse models and tested whether this Amphiphile vaccine approach could expand CAR T cells in vivo. We did this first in non-tumor bearing animals where we'd lymphodeplete them and then transfer CAR T cells mimicking clinical CAR T cell therapy procedures. And then a day later, we would either give them a vaccine boost or just leave them alone, and we would give a second injection 7 days later. And then we followed basically the frequency of the CAR T cells in the peripheral blood. And what we found is if we transfer 10,000 CAR T cells into these mice, once they've diluted into the tissues, we can't even detect them above baseline here in orange because they're too low a frequency to even detect. If we inject AMP FITC for these FITC-specific CAR T cells, just the Amphiphile molecule alone, it expands these T cells and they reach about 10% of all CD8 T cells in the peripheral blood. And then after the boost, they slowly contract, just like a natural T cell responding to a vaccine. If we mix the AMP FITC with an adjuvant, a STING agonist, to activate inflammation that will activate the dendritic cells so that when they're presenting this ligand, they're driven to display all of the co-stimulatory and cytokine support that they would normally express during T cell activation. Now those 10,000 CAR T cells are expanded up to reach 40% of the T cell compartment, and 10,000 CAR T cells look like 10 million CAR T cells transferred with no vaccine. So a tremendous proliferation of these CAR T cells triggered when we give the Amphiphile ligand plus an adjuvant, just like we would give a natural antigen plus adjuvant in a vaccine. Now, when we looked at what effect this has on the CAR T cells, we saw many changes in CAR T cell function. And now here I'm switching to the second model of stimulation, where we have a mimetope peptide that binds to a CAR that recognizes a mutant tumor antigen, EGFRV3. And now we're going to work in tumor-bearing mice that are injected with glioblastoma cells expressing mutant EGFRV3 as a target antigen. And 5 days after injection of the tumor cells, we treat with CAR T cells that target EGFRV3, and then we either vaccine boost them or not. And a week later, we analyzed what was happening with the T cells. And here what we saw is that when we gave the vaccine boosting, the CAR T cells were massively expanded in the tumor, not surprising because we know we're proliferating them. But more strikingly, when we look at the single cell level at the functionality of these cells, we see that these CAR T cells are expressing more effector molecules like granzyme, that's an important protein for killer T cells to kill their targets, and they express about 10-fold greater levels of effector cytokines like interferon gamma and TNF. And even their mitochondrial load is increased, suggesting these CAR T cells that have been vaccine boosted are metabolically more fit. And these changes in CAR T cell function translate into substantially enhanced efficacy. So now, if we repeat the experiment, we implant glioblastoma cells, we treat with CAR T cells, and then we vaccine boost or not. And now we follow for survival and tumor progression. What we see is that if we give a dose of 1 million CAR T cells that has only a modest effect on tumor progression and really no impact on overall survival, if we vaccine boost that cohort of CAR T cells, now tumor progression is halted, and about 60% of the mice will completely reject their tumors. So a dramatic enhancement in anti-tumor efficacy. And excitingly, one of the other things we observed is if we take these cured animals and re-challenge them many weeks later after they've cleared the tumor with parental EGFRV3 expressing tumor cells, well, they reject those tumors that we re-challenge them with, whereas naive mice that are injected with the same dose of tumor cells, the tumors steadily grow out. Perhaps not surprising, so the CAR T cells are persisting. But more strikingly, if we yet in another 50 days or so challenge them with EGFRV3 negative tumor cells, so the same tumor cells but lacking the antigen that the CAR T cells are targeting, these animals still rejected their tumor. And that suggested that we're inducing an endogenous T cell response that's helping out the CAR T cells. So the CAR T cells targeting EGFRV3 come into the tumor, they induce tumor cell death, dendritic cells pick up those dying tumor cells, and they present new tumor antigens to endogenous T cells that come back and help with the anti-tumor response. And we can show this more explicitly by repeating this therapy experiment: implant glioblastoma cells, treat with CAR T cells, vaccine boost or not, and then at 3 weeks, we look by ELISpot for endogenous T cells that are recognizing tumor cells. And we can see that if we give CAR T cells alone, there's really no induction of an endogenous T cell response. But if we give this vaccine boosting, there's a strong induction of these endogenous tumor-specific T cells. So this, we think, is quite important because this addresses one of the key limitations of CAR T cell therapy, which is the potential of tumors to simply downregulate expression of the targeted antigen that the CAR is going after, and thereby escaping the immunotherapy. And here we've got an endogenous response that could target additional antigens to help protect from that escape. So to summarize this part of the story, I've shown you this idea of designing Amphiphile CAR ligands that will, on injection, hitchhike on albumin to the lymph node, and then transfer into the membranes of antigen-presenting cells to stimulate CAR T cells in a manner closer to the way natural T cells are primed. And when we do this, we elicit a profound enhancement of CAR T cell activity in tumors that leads to natural antigen presentation in the draining lymph nodes, where dendritic cells are presenting tumor antigen to native T cells that come back and further bolster the anti-tumor response. And we're quite excited about the prospects of this as a simple strategy to improve the efficacy of CAR T cell therapy in solid tumors. Okay, and so in the last few minutes, I'm going to change gears and switch back to vaccines for natural immune cells. But now we're going to switch gears and talk about the B cell side of the immune response and a different setting, the setting of mucosal immunity. A lot on people's minds these days with the COVID-19 pandemic, where we'd really love to have vaccines that are effective in eliciting protective antibody responses in the respiratory mucosa, the nasal passages and upper respiratory tract. This is important not only for COVID-19, but for a variety of respiratory pathogens. But to date, we still really lack effective tools to elicit potent mucosal antibody responses. And in part, this is because it's thought that the most effective way to elicit mucosal immunity is to immunize at mucosal sites. But it's very difficult to immunize through, for example, the nasal passages where the mucus layers that line the epithelium in the nasal passages have the job of efficiently clearing away material. And so to get vaccines taken up efficiently and safely across the nasal mucosa is a pretty big challenge. Well, so what we started thinking about was using the same Amphiphile platform as a means to deliver protein antigens for B cells. And we first looked at parental immunization and found that if we attached small proteins such as a GP120 fragment from HIV envelope, it's about a 30 kilodalton protein to an Amphiphile tail. Just as I showed you with small peptides, Amphiphile proteins, when we inject them subcutaneously, they would be efficiently delivered to draining lymph nodes at much higher levels than soluble protein. And this would lead to, similar to what I showed you with T-cell responses, if we target the antigen to the lymph nodes, we get great increases in antibody responses. Here, about 2 logs in the serum IgG response to this Amphiphile GP120 versus a soluble form of the same protein immunogen. But there's an interesting piece of biology we can think about now using these same molecules in the setting of mucosal immunization. So in the airways, antibodies and albumin are constitutively transcytosed bidirectionally from the interior tissues across the nasal epithelium into the airway via a transport process mediated by the neonatal Fc receptor. So the neonatal Fc receptor binds both to the Fc domain of antibodies, and it also binds to albumin, and it's used as a transporter to transcytose these materials across the epithelium. And of course, for antibodies, this is an important means of getting antibodies into the airway, into the mucus for protection, and also to have albumin present to maintain osmolality. So we hypothesized, because this transport process is thought to be bidirectional, that this might be an effective means to get our vaccines taken up across the airways, because on binding to albumin in the mucus, maybe our Amphiphile protein could be transcytosed back across the epithelium and be delivered to the nasal-associated lymphoid tissues that lie under the epithelium in the nasal passages to drive anti-tumor immunity. And so we started testing this by making labeled Amphiphile GP120 proteins that were fluorescently marked and administering them intranasally into mice. And we saw that, lo and behold, if you administered soluble GP120 protein and then imaged fluorescent signal in the snouts of mice, you could see that there was signal there at 24 hours, but it was quickly cleared away. That signal persisted much more strongly when we gave an Amphiphile GP120, and it persisted for longer, so lasting over several days, meaning that the total area under the curve for exposure to the vaccine was greatly increased when we used this Amphiphile tail. And when we look by histology in the nasal passages, so this is taking a perpendicular cross-section through the nasal passages of mice, you could see this really striking alteration in where the GP120 antigen goes. If we give the soluble GP120 antigen in the nasal passages, it gets trapped at the top of a layer of mucus. So here's the surface of the epithelium. This is the nasal passage filled from both sides with mucus, and the antigen in red is all localized at the top of that mucus layer. Very little of it is getting down to the epithelium. But if we administered our Amphiphile protein, very little of it is in the mucus layer in between the epithelium. And instead, we see it strongly localized onto the epithelial surface and starting to transport underneath. And if we zoom in and look at higher magnification, staining with DAPI to see the nuclei of the epithelial layer, you can see that, again, the GP120 in soluble form has very little ability to even reach the epithelial surface, and we don't see any protein getting underneath the epithelium. The Amphiphile protein is getting to the epithelial layer and also getting transcytosed underneath the epithelial layer. And if we use FcRn knockout mice that can't carry out this transport mechanism using albumin as a transporter, then the amphiphile gets to the epithelial surface, but now transport underneath the epithelial barrier is blocked. So we really need this natural albumin-mediated pathway to see this effective transport. And now this leads to some dramatic effects on humoral immunity. If we vaccinate with these amphiphiles intranasally and look at serum IgG, serum IgA, we see that the amphiphile immunization elicits 3 orders of magnitude stronger antibody responses, and IgA at high levels, whereas soluble protein elicits essentially no immune response. And more strikingly, at distal mucosal surfaces like the rectum and the vaginal tract, we also see strong IgG and IgA elicited by the amphiphile vaccine that's essentially absent for the amphiphile protein. And further, we can see in the vaginal tract the induction of long-lived antibody secreting cells that persist greater than a year after immunization. So this is really a tremendous response that's persisting where antigen-specific, GP120-specific antibodies are being made in the bone marrow and the vaginal tract, both IgG and IgA, when we administer this amphiphile vaccine, but not when we give a soluble protein. And lastly, we wanted to know if this translates to large animals that have physiology of the nasal passages closer to humans. And so we carried out studies in rhesus macaques. First, we gave fluorescently labeled protein intranasally into macaques, and we measured uptake into the nasal tissue. And similar to what we saw in mice, there was greatly enhanced persistence of the antigen in the nasal tissues. And then if we immunized repeatedly in macaques and looked at the serum IgG and IgA responses, we could see that especially after a single immunization, all of the macaques start making IgG when we give the amphiphile immunization, but it takes several boosts to get that response up in the soluble protein. And even after boosting, the amphiphile protein elicits a response that's about a log higher than the soluble protein vaccine for both IgG and IgA. And importantly, we also see enhancements in the IgG and IgA response in the nasal passages. And this is something we think could be quite important, especially for respiratory pathogens. And in the last thing I'll show you, let me just show you that, of course, as with many vaccine groups, with the onset of the pandemic in 2020, we immediately sort of pivoted to ask, could this technology be helpful to make more effective mucosal vaccines against SARS-CoV-2? And so we made an amphiphile RBD immunogen for SARS-CoV-2. And similar to what we saw with our HIV antigens, if we administer these vaccines intranasally, we get strong serum, vaginal, saliva, nasal, and bowel IgG and IgA responses, where soluble RBD protein elicits very poor responses, especially in the nasal passages and especially for IgA. And if we measure neutralizing antibodies, there's really no comparison. In the nasal wash and in the bronchiolar lavage, we can see strong neutralizing antibody responses with an amphiphile RBD immunization that's essentially completely absent with soluble RBD protein. So to summarize this part, I've tried to show you that using these albumin-binding immunogens is also a way to enhance mucosal immune responses. Again, just exploiting the natural biology of albumin, which is naturally trafficking bidirectionally across mucosal barriers, and using this as a way to promote humoral immunity at these mucosal sites. So I'll stop there and just thank again several really talented members of the lab. The work on the amphiphile vaccine was started in the lab by Haipeng Liu, and then carried on in the CAR T-cell story I told you by Leo and Ma. And the mucosal immunization I showed you was all the work of another talented postdoc, Brittany Hartwell. And we're grateful for support from the NIH, the Mark Foundation, and the Marble Center for Cancer and Nanomedicine on the work that I showed you here. So I'll stop there, and I'd be, of course, glad to take any questions if there's time.
Thank you, Professor, for your wonderful talk. Does the audience have any questions? Yes, Shalu ma'am.
Great talk. I had a question from the first part of your talk where you talked about the CAR T therapy for the solid tumors. So the currently established, the FDA-approved CAR T therapy for the hematological malignancies, we see the cytokine release syndrome, the cytokine storm. That's one of the problems that is there. So I was wondering if you could comment on if you have tested that and if you have seen like how the cytokine profile looks like in the CAR T cells that are proliferating in vivo in your in the system you have developed.
Yeah, that's a great question. Yeah, we did look at that quite carefully, and not only for the concern of CRS seen in the hematologic malignancies, but also because if any of these amphiphile molecules were to get into the systemic circulation, there'd be concern they would stimulate CAR T cells and who knows where. So we've looked at that carefully, and I think the key finding there is that when we parentally administer these amphiligans, they go to the draining lymph node chain. They get essentially completely captured in the first couple of lymph nodes in the chain, and they don't get any farther. And so that stimulation is being confined to the lymph nodes, and that seems to be very safe. So we don't see CRS or other signs of liver toxicity, for example. So we're encouraged by that.
Thank you.
Thank you, Shalu ma'am, for your wonderful question. Does anyone have other question?
I can ask one.
Yeah, sir, please go ahead.
Darrell, wonderful talk always. I enjoy your work. So in the last part where you have the SARS-CoV-2 response, so have you looked into the memory response in that and the localization of the cells that are being generated in some of the lymphoid tissues?
Yeah, that's a good question. We've only we measured memory cells in the blood and spleen just to see if we were changing the magnitude of the response, and we do see more memory cells. But we haven't done the same kind of dissection to, for example, look at whether we're getting local plasma cells or memory cells in the mucosal sites. One of the hard lessons we learned doing these experiments is I think mice do have different biology than macaques and presumably humans for these tissue resident responses. We saw just tremendous what people call common mucosal immunity with the amphiphile immunization in mice. As I showed you, you could get every mucosal compartment making IgG and IgA, and we did see evidence for local plasma cells there or antibody secreting cells. But in macaques, what we found was we only got the local mucosal response. We got in the nasal passages and bowel, but we saw pretty weak and sporadic responses like at the distal vaginal tract and the gut. So it's made me think that if we're going to look at that we should look at it in macaques because it seems like things are different in mice, or it could be that a different adjuvant is needed in macaques. But yeah, that's what we saw.
Yeah, I think that's an important point because there are some studies. I think there was one study from Emory that came out in Science Immunology for SARS-CoV-2 in the macaque study that showed that with the TLR7/8 adjuvant, the plasma cells were not really leaving the lymph nodes, I believe, which is not what we had seen in a lot of other preprints and other studies that came out previously. So.
Yeah, that's right. And that's been seen with some HIV vaccinations too. So yeah, some interesting differences as we get to the larger animal.
Okay, thank you.
We have a question from Veer. Can you ask the question or shall I read it to you?
So could you kindly expand further on the efficacy of the Amphiphile boosted CAR T cells in eliciting a response against both EGFR V3 plus and EGFR V3 minus?
Yeah, so what we've seen is that in the setting of where we vaccine boost them, the CAR T cells become capable of promoting endogenous T cell priming. And I did, for sake of time, I left out. We've studied this in detail now, and what happens is when they're vaccine boosted, the CAR T cells are metabolically more fit, and they're able to sustain their interferon gamma expression better in the tumor, and that makes them better killers. And so there's a lot more tumor antigen being released, taken up by dendritic cells and carried to the lymph nodes to activate new T cells. And in fact, this response is potent enough that if you make mixed tumors where you have, for example, 20% of the tumor cells lacking the target antigen, the EGFRV3, at time 0, the CAR T-cell therapy will still clear the tumor in about 40% of the mice, because the endogenous immune system comes in and helps clear away those antigen-negative tumor cells. So we think this could have pretty significant implications for human tumors where, especially in glioblastoma, the EGFRV3 expression is known to be very heterogeneous. One patient might have a lot of EGFRV3 expression, and another patient only a part of the tumor cells will express it. So we think this could be an important way to make CAR T-cell therapy more effective in those instances.
Thank you, sir.
Thank you, Professor, for your wonderful talk. We'll go to the next speaker soon. Have a good day, Professor. Thank you.
Thank you.
Ladies and gentlemen, our next speaker for the day is Dr. Robert Chang, who joins us from Stanford University. Dr. Robert Chang is a fellowship-trained refractive cataract and glaucoma specialist working at Stanford University since 2009. His research team harnesses glaucoma insights from institutional big data comprising electronic records, imaging database, and a growing biorepository. Previously, he helped in developing the normative databases for ubiquitous optical coherence tomography, OCT. Currently, he has been interested in privacy-preserving computational methods, predictive analytics, and testing novel portable medical devices, that is, virtual reality headsets and small molecules. He is a formally trained in biodesign, biopharma, and process improvement science. Dr. Chang co-invented the iHo smartphone imaging adapter, which was licensed and commercialized as the Paxis-Scope. He previously co-founded a venture-backed seed-stage consumer health startup and is a scientific advisor to multiple Fortune 500 and small pharmaceutical, medical devices, and digital health companies. Ladies and gentlemen, please join me in welcoming Dr. Robert Chang.
Welcome, Professor. Would you like to share your screen for a presentation?
Yes, is it working right now?
Yeah.
How about there?
Yes, it's working. It's
Okay.
on full screen.
Good evening. Thank you all for staying late. It's a privilege to deliver a keynote in honor of late Professor Banerjee. I'm an ophthalmologist at Stanford University. So why am I here speaking to the Nanomedicine and Drug Delivery Group? I have never studied any of those domains. I think this talk will be a little bit different from your other talks. Some of you may remember the Think Different ad campaign from Apple, one of the most recognizable and innovative brands in the world. During my short time with you, I hope to demystify the process of medical innovation. I always thought you had to be at the right place at the right time to be a successful innovator. But sometimes you just need to be different than who you studied to be. While I'm an ophthalmologist by training, I am not your typical medical doctor. Professor Rinti Banerjee was someone who wore multiple hats before her life was tragically cut short. She was named the Madhuri Sinha Chair Professor at the Department of Bioscience and Bioengineering at Indian Institute of Technology, Bombay, India's top university. She was a renowned nanomedicine leader, star physician scientist with multiple innovations and inventions, an amazing mentor working across multiple disciplines from neonatology to chemical engineering. I wish I had an opportunity to meet her. So how many hats do you wear? Some of you may be a PhD or a medical doctor. I would argue that more hats you earn, the more successful innovator you may become. It's hard to wear every hat, however, such as like a chief executive officer, CEO, chief technology officer, chief medical or marketing officer, because of all the differing skills required. But entrepreneurs often have to start with multiple hats. As for myself, most of the time I wear the hat of an ophthalmologist who sees patients and performs eye surgeries 80% of the time. As a university professor, I'm also responsible for training the next generation cataract and glaucoma specialist. My job requires publishing new research findings, but I also really love wearing the hat of innovation and startups, where my impact can be multiplied by bringing new technology to many. I've been trained in biodesign and implementation scientists. I'm a medical director of Stanford's employer-based optometry. I'm former vice president of the Asia Pacific Teleophthalmology Society, focusing on AI and big data. So I've leveraged all this experience and business acumen to consult for many companies in biopharma and med device. And my final hat is in global health, where I traveled to many countries around the world as a visiting professor, performing research, spending sabbatical time, testing startup products, volunteering to teach surgery, and just learning about different medical systems. And all of this has been encapsulated as an academic physician. But actually, before all those hats, when I first arrived at Stanford, I thought innovation had to just do with getting lucky. Maybe you had a big idea and you happened to be with the right people at the right time. It's kind of a big black box for me. There was maybe throw in some magic and the invention happened. But here I was surrounded by many successful entrepreneurs within the Stanford community. If they can all do it, why can't I? Early on, I really learned that the most common mistake in medical innovation was trying too hard to use what you know. It's like taking an existing tool and keep trying it on different scenarios until it, quote, works. While this is taught in terms of pivoting and never giving up, in reality, for medical innovation, this is very inefficient and costly. Just because you have a big idea doesn't mean it's the best solution for the problem. So don't be the hammer looking for a nail. In fact, a design innovation lives at the intersection of these three areas: the human value or usability, the technology or feasibility, and the business or viability. It's important to point out that not every innovation needs to be a business, but is more likely to be sustainable as a business. And the design thinking process is a series of steps, which is usually used for non-medical things, but it can also be used for medical innovation. If you think about empathizing with the patients or the healthcare workers, defining the problem, ideating, prototyping, and testing to reach that central design innovation sweet spot. So for healthcare, where should you start? Well, really, before brainstorming any solutions or trying out any hammers, it is important to always start with identifying the need. We all have limited time and resources, and medical inventions can be expensive and take a really long time to reach patients, sometimes like 10 years. The US has a very strict FDA regulatory environment for drugs and devices, thus always trying to characterize the unmet need first to make sure it's worth the time and resources is important. You really can't just think about this. You can't do this in your mind. You must get access to patients and healthcare workers to really understand the need. For many needs, through direct observation and research, you can typically narrow down to the top need based on specific criteria, including your team's expertise. Only then do you get to brainstorm the many potential solutions before narrowing down to a top solution. And then finally, you can try to de-risk the pathway of your big idea all the way to commercialization if you decide to start a company. So you might be saying right now, I can't do all that. This is why you need a large professional network. In addition to the big idea, you need 1, a team with diverse set of skills, 2, a product market fit which adds value and can't be copied by fast followers very easily, and 3, some funding and connections to set milestones. It's very important to ask yourself, how much do you believe in your idea? How persuasive are you to convince other experts to join you? For healthcare innovation, identifying, inventing, and implementing can look like 3 totally different people. You could spend a third of your life obtaining 3 degrees, or instead team up with medical, technical, and business experts. For example, if you are the medical expert like myself, you tend to need a technical expert to visualize and test your ideas. If you're a technical expert, you tend to need a medical expert to help make sure you understand the problem you're trying to solve. And if you're a business expert, it's best to join a founding team who has an unmet need and potential solution, but really needs help with forming the business. Where do you find all these people? Silicon Valley is an opportunity-rich environment for innovators, just like Hollywood is an opportunity-rich environment for entertainers. And by the way, Silicon Valley show is surprisingly accurate characterization of what goes on around here. One of the unique things about Stanford is that the School of Medicine, the School of Engineering, and the School of Business are all right next to each other, all on the same campus. And this encourages a lot more cross-pollination. So for the beginner inventor, I think the most important thing to remember is always to start with the unmet need. Don't be a potential solution in search of a need. It can be costly and inefficient. So here's what Thomas Edison had to say about this. I find out what the world needs, then I go ahead and invent it. He was a good inventor, but a better need finder. Dr. Polyak, the director and author of Biodesign textbook, emphasizes, quote, a well-characterized need is the DNA of a good invention. It's a lot of work to get the need right, but once you are there, the invention will almost certainly follow. There's a systematic approach called the biodesign process, which helps make sure no stone is left unturned during your entrepreneurial journey. So let's look at a basic example of characterizing a need. What if you were a scientist back in the 1900s, you were very observant, and you thought, you know, here's a problem. I wish my horse isn't so slow and wouldn't get tired so easily. If that was a problem, you may start thinking of ways to get the horse to go faster. But what if the underlying problem was really a way to transport people quickly and reliably? With the problem stated that way, you may come up with a car instead of a faster horse. And this is why scoping a need is very important. It helps prevent being too general, like the need is just to try to cure cancer, or being too narrow and not being generalizable. Oftentimes, someone invents something because their family member or they themselves are a patient. But if that's not the case, you need to talk to a lot of patients, a task which falls under what we call customer development interviews and research. You really need to know who your target users are, just as much as you know about nanomedicine and technology, gene therapy, CRISPR, CAR T therapy, all of that. Oftentimes, you actually may even gain insights from patients themselves who are trying to work around their problems. Maybe you see how they deliver eye drops in their eye, for example, and you decide to make a device to facilitate that. Unmet needs are also analyzed in the context of market size, cost, disease state fundamentals, really understanding the pathophysiology to make sure the science is well understood. Comparing the treatment landscape with a gap analysis to see what the alternatives are, and a stakeholder analysis so you can understand how the new potential solution would affect the industry and who may actually want it or who may actually try to block it. So with limited time, what can you do? Well, one of the key points I have for this lecture is that you should try to develop a T-shaped resume with breadth and depth in a single area. As a doctor, I have a lot of depth in clinical medicine, but I actually spend a lot of my hours learning different engineering tools so I can learn how to communicate with other people who make things. I also study a lot about business so I can talk about finance and understand what is actually a sustainable solution. One example I know is actually how to CAD, computer aided design. I know how it works, but I don't try to draw anything on my own since that would take too long. So What you need to know is just the language to be able to communicate with the experts in those other areas. Point 2 is actually how you can be different. You should be curious and learn skills outside your comfort zone. I know all of you listening to this talk are really experts in nanomedicine and drug delivery, experts in COVID, all of the things that we really need scientists to be experts in. And that's your 10,000 hours, to become the expert. But what about the 100 hours to be able to talk to different experts, like talk to healthcare administrators or talk to physicians, nurses, pharmacists, optometrists, all the ancillary providers. Also, there's this concept of ikigai for work happiness I'd like to emphasize, where you try to combine with what you love, with what the world needs, with what you can get paid for, and what you're good at. If you happen to find that, that is pretty awesome. But kind of working in those areas especially can help you on your journey to making huge impact and feeling very happy. Finally, point number 3 is maximizing your environment. I founded a digital health wearable air pollution startup with 2 other Stanford faculty co-founders, one who is very good at marketing ideas and the other one who is very good at technology and making things. I helped get the day-to-day things together like a COO. Therefore, the 3 of us formed a very complementary team. We were able to raise angel funding and venture money together. And from that experience, I really learned how it takes the right set of people together to sell the vision so that others can support and help make your dream possible. Well, I want to take you back a decade or so now when I was working in the eye clinic and the first iPhone cameras were starting to rival point and shoot cameras. I was trying to just be able to take pictures of the eye with the iPhone. This video shows a friend trying to line up the camera with our slit lamp microscope to provide a magnified view of the eye. There's also portable versions that we were using for eye examinations that we just attach point and shoot cameras to. And as an attending doctor, young doctor at Stanford, I was covering call and I noticed a lot of my residents, trainees were always texting photos to try to get quick answers while they were seeing patients in the emergency room. But oftentimes these pictures were just too blurry or too small, and thus I characterize an unmet need as a way to quickly capture eye photos to share securely among healthcare providers to improve remote triage. So what did I do about that? Well, I couldn't build anything myself, but I attended a Stanford hackathon event where I pitched this idea of trying to improve the smartphone to capture pictures of the eye. And I met a wonderful group of Stanford students of various backgrounds who joined me, and we almost actually won. But at least one of them stayed in touch with me even after the event. And this was the beginning of IGO. There's a paper around the same time which spurred my excitement from a team in Boston who published that they were able to get pictures of the retina using their existing ophthalmic lenses and an iPhone, a technique which requires a lot of skill to hold things steady at the right distance, in order to capture a clear image. So me and a Stanford Master's Engineering student from the hackathon and a Stanford resident, MD, PhD, worked together to create a minimum viable product, MVP, called IGO, using a dorm room 3D printer. Well, it doesn't really get more Silicon Valley-esque than that. We needed pictures of both the front of the eye and inside the eye and the retina, but the easier task was to get pictures of the outside of the eye first. So we just started playing around with some macro lenses and different lightings to try to capture images of the cornea. We iterated really cheaply, just put some Legos together and LED lights, putty, used some clips, until we kind of met the design criteria of we need something that was really small and portable, something that was really fast to attach and detach or people wouldn't want to use it. Next, we worked on attaching the lens to the phone, as humans don't have a third hand yet. This involved multiple iterations until we started 3D printing different designs to get the camera alignment right. Every time we thought we had made progress, we ran into another problem. Nobody would want to carry a bulky thing in their white coat pocket, so we came up with this foldable design. Each smartphone camera had a different light with a variable location and control, so we had to make our own standardized light source. None of this could have been solved with just thinking about all those variations. All the insight came from experimentation. We were obviously passionate about this project, which we did on the side, and we started gaining a little bit of traction. We took the adapter to international rural settings where there were no cameras, and the medical cameras that did exist cost tens of thousands of dollars. So here we had a value, a cheap portable device that could take pictures of the eye, and that was immensely useful in the field. We even performed some eye screening camps in India with the help of Stanford research students who traveled out there. We also won some grants, which provided funding to get an app programmed because we needed a way to store the images. And then we started collecting them through IRB's studies. So here's some example pictures of the front of the eye. Mind you, of course, this is many years ago now, and obviously we can do these things even easier with our current phones. Here you can look at the detail. See the blood in the right of the eye on the upper right, or maybe the white spots on the cornea on the lower left, indicating a corneal ulcer infection. Picture on the lower right is a corneal transplant, and this type of information is very useful during a telemedicine triage. Dilated retinal pictures were even more striking, where we could identify and document serious conditions, such as hemorrhages in the eye. Stills were extracted from those videos, and this was great for teaching as well. These tools were facilitating bedside image comparisons for better continuity of care, and many eye institutes in India were doing this at the same time. So we ran a clinical study with the device in our county hospital looking for retinal hemorrhages, which is something that we do as a screening for diabetics already annually. Our device was able to capture moderate to severe retinopathy, a clinical meaningful endpoint in which treatment is instituted with a kappa score of 0.9 correlation. So now our little invention was not just something cool, but it actually went on to be published. And this is how you really gain back time for all those side projects that you're working on. It counted towards publications for promotion. And after looking at the market, regulatory path, reimbursement, team, etc., we decided it was better to license the technology to actually start a company. And I learned so much from this process. But nowadays, these smartphone cameras adapters even have built-in AI software to perform automated diabetic screening. So I think one of the key elements of implementation, after identifying and inventing, is to look at the business model canvas. It's a classic resource to help startups figure out a scalable and sustainable business model. Not just to think about the profit, but business is something that can be learned by doctors and scientists alike, and is one area I recommend spending a lot more time on, especially those extra 100 hours. So now let's recap the medical innovation process with some of the magic unveiled. First, we start with a pain point, and you try to characterize the unmet need so that we know the market, we know the disease fundamentals, the competitive landscape, the stakeholder analysis. Once the unmet need is chosen with need specifications, we can iterate some potential solutions to reach product market fit with customers. We figure out about the business angle to make it a sustainable venture and figure out how to protect the business against fast followers. Finally, we define the cheapest minimum viable product to gain some traction and start pitching for additional funding so that you can hit certain milestones. And simultaneously, we've recruited a strong, diverse team which will use that funding like pouring fuel on a fire to accelerate your progress. So let's reflect for a moment for those of you who are a nanomedicine student. Probably means you have really deep science expertise. You probably have a topic of specialization that's like your hammer. But have you ever thought about unmet needs first? Maybe a plan to grow your breadth of knowledge? Spend some time thinking about how to move some of your ideas forward that you've always wanted to try but didn't have time. Maybe you've studied a little bit about regulatory pathways and reimbursement strategies because biotech is very specific knowledge different from medical devices. So in summary, I'd like to think that this presentation introduced you to something different, something you wouldn't hear from a typical ophthalmologist. So some 3 driving principles I leave with you are: 1, to develop your T-shaped resume. 2, to maximize yourself by being curious and going outside your comfort zone into areas that are maybe not in the science realm. And 3, how to maximize your environment, how to increase your network, how to meet the right people who are going to help you in your journey of entrepreneurship. Maybe you need to work with more doctors or more business people, because oftentimes, even for myself, I'm always in the healthcare environment. I'm never really hanging out with a lot of engineers or business people unless I do it by myself. So try to find those other like-minded entrepreneurs. I will close with a favorite quote of mine by Daniel Pink. The three things that motivate creative people: autonomy, mastery, and purpose. For anything you spend a lot of time on, make sure you have those things. And if you didn't catch Ikigai before, remember to combine what you love with what the world needs, with what you can get paid for, and what you're good at. Thank you very much for your attention, and I'm happy to take some questions now. Sorry, you're on mute. Okay.
Yeah, sorry, I was on mute. Thank you, Professor, for your different presentation in the regular scientific talks. I enjoyed a lot personally, and you somehow reminded me, Professor Banerjee, in some way. I will request participant if they have any questions or comments, please go ahead. Yes, Ekta, please go ahead.
Hello, Professor.
Hello.
As Saurabh said, equally you reminded me of Professor Banerjee. She used to always think about the different scenarios, the stakeholders, and about the industry requirements, and So I think this was. I really like to mention that this was an appropriate talk to end for all the students, for all the young budding scientists that have attended this conference, and I think this was the best way to end the session today. I have one question actually. So, so the hello.
Yes.
Yeah.
I can hear you.
So yeah. So I actually wanted to know that the device that you had made, the IGO device,
Yes.
and did you ever think on repurposing it for imaging of some other, like, I know that you are from ophthalmo, like you're an eye specialist, but like, did you ever thought about repurposing the device for some other kind of imaging?
Yes. At the same time, when we were working on the eye version, there were other Stanford students who were working on the skin version, for example, like a dermatoscope. So there was a lot of on how to standardize like measurements of the size, the color. There's actually a lot that goes into imaging, actually. You think a picture is just a picture, but when you actually use it to grade medical things, you have to create a lot of standardization. And so certainly there are different groups working on different angles. And because I'm, the eye specialist, I was working mostly on getting useful things that were properly in focus for the eye, for conditions that relate to the eye. But yeah, you're right. This is another way to innovate, is to actually think about adjacent areas that someone else might not have thought of, that you can use your great idea.
Okay. Thank you, Professor.
Anyone and wanted to ask question or add something? So, Professor, I just wanted to ask, like, do you think doing a design thinking course actually helps us, or like, it just adds on the CV, really?
Depends on how much you know already. Certainly, you can read things in a textbook, but generally when you work in groups and do hands-on activities, I think it solidifies the principles a lot more. Stanford offers like a Stanford India Biodesign, like 6-month fellowship kind of thing, where you spend half time at Stanford and half time in India, and working together with experts on how to deliver your potential idea. And there is a lot to learn that's different from, let's say, like I presented something sort of digital health, medical device related. It was a class 1 device, relatively easy to get regulatory approval. I wanted to make it very simple like that as an example, just to kind of show you the process. But certainly, I imagine a lot of your ideas, maybe, class 3 or something PMA, something definitely that, you would need a lot more expertise than just like reading a book, because the complexities of a regulatory environment, generally you're hiring experts in those areas just to understand what needs to go through. Like, let's say you had a new molecule or using a nano robot to go inside the bloodstream to repair something inside the body, like science fiction, so to speak.
Right.
Well, you would really benefit from going through courses that break down all the different elements of why we have, infrastructure set up the way it is to make these medical things, so complicated. Why is it so hard to test that kind of stuff in a human? How do you get to first in human? What about the safety? What efficacy endpoints do regulatory bodies care about? All of that, the more you immerse yourself, the more you take courses, the more you speak to other people, you get better and better at over time. And that's why a lot of healthcare innovations, they don't just happen when you're like 20 years old. I mean, these things are happening in much more senior people who've been around, who've seen the development life cycles that take a long time, 10 years to launch products. And then they're able to help the new great ideas, come to pass. So I definitely recommend taking those biodesign-like courses. It doesn't have to be Stanford Biodesign, by the way. There are other courses as well. I happen to be at Stanford, so definitely, I'm fond of this. I took it myself, but there are like Hopkins, like a lot of places do innovation work. Texas Medical Center.
Right, because I attended one short course which offered by one of the Stanford professor in IIT. So it was sponsored by IIT Bombay, but I just attended. So I really thought of taking that course further ahead and seriously. But I really wanted to know, and it might help my other colleagues who were attending the today's session to know that this really helps or not. But you answered my question almost.
Yeah, one last point I'd like to say is that I think you can do those little certificate courses to get the introduction to see if it's interesting to you at your stage of life, at your time.
Correct.
Depending on what you're learning already. And thus, you should only go for the full, year-long thing if you're definitely committed. Because a lot of those people, maybe they're in transition, they didn't really like what they were working on, they want to start something new, then that's perfect opportunity to make the switch.
Correct. Okay. Anyone wanted to add comments or something, or shall we just?
I think Abhinanda has a question.
Oh yeah, Abhinanda. Sorry, I didn't see her.
Abhinanda, please go ahead, unmute yourself and ask the question.
Am I audible, Professor?
Hello.
Yeah. Hello. So I have a very nice question for you. That is that you have said that we need to identify the unmet need and then we go ahead to address that with our innovation. So identifying, we might be able to identify something, but nowadays, like with the current advancement in technology and science, once a need is identified, we'll find maybe hundreds of groups working to address that need. So how do we sort of identify that what area we should address that will set us apart from how other people are addressing that need? So it is in this tough time of competition that everybody is running after maybe a single problem is being addressed by thousands of groups across the world. So just a bit of insight into that, like how we can identify how to go about
usually, in healthcare, identifying the unmet need is driven a lot by the person who's treating the condition. Let's say you're targeting some kind of disease. If you're not actually taking care of the patients day in and day out, it's hard to see something that's very subtle or different than what everybody else is seeing. So if you really want to be like game-changing innovation, usually there's an observation of something that's happening that other people aren't seeing. Because if you're seeing the same observation that everyone else is seeing, that's usually more in the like fast follower realm. Someone else identified the need, and many people are trying different solutions simultaneously to see which one's going to win out. And sometimes there's some business factors that make one win over the other. Or some timing factors, or a lot of like maybe some things not even under your control. But I think as long as you're in the realm of working on the unmet need to begin with, and you've characterized it together with other healthcare workers, that puts you in the best chance to succeed and move forward and gain additional funding to keep working on your idea so it's more differentiated from the competition. I like competition. I think that means you're working on a need that's very important. If there was no competition, it's probably not necessary. So don't be afraid of competition. I think it helps, make everything better as always. But usually there's an angle of your view of the problem that is differentiated from someone else if it's a true sort of novel thing. And that may be some like scientific paper or some new timing thing that worked now that didn't work long time ago, something that, you know, or it's in a different environment, like, okay, those people are doing it in the office, but you're actually doing it in the home setting. So something very, very different that helps you stay ahead of the competition. Certainly, you know, the more you're trying, I think you'll see that there are ways to be different from the competition.
Thank you so much.
Yeah, thanks, Abhinanda. So we'll be concluding this track. I want to really thank Professor Molly, Professor Ankur, Professor Nicholas, Professor Darrell, and Professor Robert for wonderful talk today. I hope everybody has enjoyed and got some insights. Actually, I was thrilled right now by listening to these different talks. And by this, I would like to conclude this session. I request all participants to see the chart and click on the links which is pasted for the validatory session. I hope to see you there soon. Thank you.
Saurav, we are going to say in the same track. This is the first track, so the validatory session is going to
be in this track, please.
Yeah.
No,
Yeah.
sorry, Ekta, sorry to interrupt you. So
Yeah.
join the link that Saurabh shared in the chat box. That is the link for the validatory session.
Okay.
So requesting all of you to join us for the validatory session, and please join the link that Saurabh shared in the chat box. Saurabh, can you please announce again it?
Yeah. So it's already announced, and everyone must have heard.
बंद किया रिकॉर्ड हो रहा था बंद हो जाता है कॉल पे बंद
So Saurabh.
Bolo.
Yeah. So Saurabh, just please join the link that you shared. So I'm going to end this meeting for all.
ठीक है, मैं ज्वाइन करता हूँ बाकी के लोगों के मीटिंग के लिए. हाँ?
Sure.
Thanks.